My name is Wendy Vanasco, and I am a project manager for the Polycystic Kidney Disease Outcomes Consortium at the Critical Path Institute. And I would just like to welcome you all to day 2 of our 2021 PKD Regulatory Summit. Hopefully you were able to join us for day 1 yesterday. We just want to welcome you. Thank you so much for being here. We have some really great information to share with you today, some very important information. So thanks for being here. But before we kick off the meeting, I am going to go over a few housekeeping items as the meeting progresses. So just so all of you know that all participant lines will be muted during the call. So please use the chat feature if you have any comments during the event. Second, once we get to the panel discussions, please submit all of your questions to the panelists through the Q&A function, which you can find at the bottom right of your screen and at those 3 ellipses right next to the chat feature. Also, just want to mention that the workshop is being recorded and will be available on the CPATH website soon after the event. So we'll alert everyone when that is available. Last note, if you need any assistance at all during the meeting, please feel free to contact my colleague Kitty Boji or myself, Wendy Vanasco. Our emails are listed here. So if you have any technical issues, questions, please let us know. We'll be happy to help. And with that, I am going to turn this over to John Michael Sauer. Let me give him presenter control here. So John Michael is the Senior Vice President of the Translational and Safety Sciences Program here at the Critical Path Institute. He is also the Executive Director of the Polycystic Kidney Disease Outcomes Consortium. So I'm going to turn this over to John Michael to take us off through the meeting here.
It's a really good discussion. So what I'd like to do is start off by a couple thank yous. So thank you to the PKD Foundation for sponsoring the Regulatory Summit, as well as Atsuka. Without their sponsorship, I don't think we could have done this summit and had it as robust as it is. You know, sorry this is a virtual workshop. I can't wait to see everybody in person, but I think we all feel that way. So as soon as the pandemic decides to let up, I think we can get back to normal and have regular type meetings and be able to chat with each other as well as enjoy, you know, the presentations and formal discussions. Special thanks to our planners and organizers, Wendy, Kitty, the CPATH staff for you know, pulling this all together, and likewise to the planning committee, Ron, Chris, Frank, and Elise, for working for these last several months. We were talking about that before the session started, but I guess we started working on this 6 or 7 months ago, trying to pull together the agenda and figure out the speakers and all those pieces. Special thanks to our speakers and panelists, of course. But let me go through day 1 really quickly, which happened yesterday, right? So we had some great discussions about AR and AD, PKD. I think we see that there is a lot of work to be done in the AR space, where it's much more, you know, almost in that basic science area, right? Where we have a lot of work to do to define regulatory pathways, to be able to enable drug development to really happen around that disease. With AD, PKD, I think we see that we've made pretty significant advancements over the past several decades, and we need to continue to make advancements, but I think what we have is a very firm grounding that we've created to be able to advance our next generation of ideas. And so again, thank you to the panelists, thank you to the speakers for the session yesterday. Looking at today, I'll be leading along with Frank a session on data-driven drug development tools. And what we hope to do here is really have a nice discussion around where we can go next, right? And how do we go there as far as, you know, drug development tools and different tools that are needed to accelerate therapeutic development. Then in our next session, 3B, Frank, Elise, and Romaldis will be leading a discussion around creating regulatory standards for outcomes for AD-PKD. I'm really excited about 3B, to be truthful, because I think it's going to be a unique discussion, because I think we can really dig into the weeds around, where we can go with these clinical trials. And so really excited about that. We know that as drug development sponsors bring their programs forward, a lot of that information becomes proprietary and is not shared. So I think we've created a mechanism here to share some of these ideas in an open audience, so that not pointing to any specific compounds, but thinking about what can actually be, what approaches can be used within clinical trials. So looking forward to that. So again, our stakeholders are pharmaceutical and biotechnology scientists, academic scientists, health authority scientists, patients and their families. So we have a very diverse stakeholder group that's attending the summit. I think one thing that we really need to realize about the format of this summit is it's not a formal binding meeting. We have regulators here. We want to hear what the regulators think from a scientific standpoint. Likewise, we have industry members here, and they're not representing their individual companies per se. Instead, they're speaking as scientists. And so what we want to do is really lower the guard here, if you will. So realize that the individual scientific opinions are not necessarily the opinions of their employer, organization, committee, or any other group that they're associated with. And so let's have a good, robust scientific discussion like we did yesterday. As I said, we've made some significant advancements over the past several years around the science of PKD, and I think we've been able to really refine and redefine the drug development process for PKD therapies, and I think we have more to do. And so I think one thing that we've really learned is that we need strong leadership around any disease area, and I think the PKD Foundation has provided that. And I also think that the Critical Path Institute has helped to drive many of these tools and ideas forward, and I know that both groups are going to continue doing that into the future. Now, we all have individual stakeholder roles to play, and we're going to play those, but I think what we know is that we all have a common vision, and that's improving the lives of patients with polycystic kidney disease, and that's what brings us all together. And so without further ado, I'll basically introduce our first session, which is going to be session 3, the future of PKD drug research. The first subsection within section 3.3a is driven drug development tools, or I'm sorry, data-driven drug development tools. And I'm one of the moderators for that, and so is Frank. And that's a heck of a picture, Frank. I'll tell you. I feel pretty confident after looking at that picture and knowing you're my co-moderator. So look forward to this discussion. What I'm going to do is first work through the session, tell you a little bit about how we're going to speak through this session. I'm going to give a very short introduction. Following that introduction, Carrie Maxwell will give a 15-minute presentation as a patient advocate, talking about her disease and her experience. We'll then have a panel session and discuss several topics that were covered by the presentations. And here are a listing of the presentations. And what you can see, I'll go through those presentations in a moment, but special thanks to all the presenters. But what you can see is for these presentations, what we really wanted to do was go ahead and, number one, identify the opportunities that we have within this space to bring forward additional drug development tools and solutions to basically facilitate the development of new therapies in PKD. What we also wanted to do was really focus on the mechanism to make these tools happen, and that's through data sharing. And how do we really generate the data that we can then share with regulators so that they can accept these tools, right? And then the ultimate objective is to go ahead and then seek regulatory endorsement for these drug development tools for PKD. And we've concentrated on three main areas: TKV, biomarkers, total kidney volume, clinical outcome assessment measures, and data sharing. And when we look at those talks, that's exactly, where we went. We have a session that was on TKV, probably the most advanced biomarker, imaging biomarker for use in polycystic kidney disease. We touched upon clinical outcome assessments in AD-PKD, and then we had two talks on data sharing. And let me just really briefly summarize those talks for you, just so for those that weren't able to see all the talks, but definitely when you get a chance, please go back and look at those talks. They will be on the CPAP website, and we'll be sending out links after this after these sessions so you can go back and look. So both Vincente as well as Klaus talked about TKV, and I think the starting point for both of their talks was really around the fact that TKV is a prognostic biomarker for polycystic kidney disease, at least the autosomal dominant type. And the talks went in two different directions. Vincente went in more of the advancements of the technology of total kidney volume, speaking to texture analysis and cyst counting, how to basically increase the amount of data that could be gained from this technique. Klaus, on the other hand, went down more of the modeling and the direct application of total kidney volume from a prognostic biomarker to a reasonably likely surrogate biomarker, and how basically modeling, computational modeling, can go ahead and basically allow that reasonably likely surrogate endpoint to be applied in drug development programs. Both Dorothy and Bex talked about the clinical outcome assessment. And I think what we saw from their talks was, number one, it looks like a pain and discomfort scale is an opportunity that we could bring forward to be used in clinical trials for PKD. Likewise, Bex showed us a pathway by which we could go ahead and develop, modify, and revise the current AD-PKD PDS scale. And I think we're pretty lucky because CPAP has a lot of experience in bringing these types of assessments and instruments forward. So I think that will make a very robust discussion as we move forward, whether or not this could be an opportunity for stakeholders to bring forward. Finally, there was a talk, there were two talks, one by Jeff and one by Leslie, around data sharing, and two very positive examples of how data sharing can happen. Jeff really concentrated on a program within CPATH called the Rare Disease Cures Accelerated Data Analysis Platform. Pretty big title, but we call it RDC-ADAP. And really what that does is pull data together around the number of rare disease. And as many of you who've participated in the PKD Outcome Consortia realize, we've been collaborating quite a bit with RDC-ADAP for housing our data and analyzing our data. Leslie talked about the CKD EPI, or the her collaboration that she's pulled together for the past several years, looking at how to share data across chronic kidney disease, specifically around looking at change in EGFR across time, as well as albumin in the urine, and looking at how that can be used as a surrogate endpoint for these various diseases encompassed by chronic kidney disease. So I think in both cases, what we saw there were some similarities about their approaches, and looking at how to best interact with investigators to share data. And then I think in both cases, that relationship that has to be built between data contributors and data consumers is very important. So again, if you didn't have a chance to review these talks, they're still available. They're all very good talks, and I think you'll learn a lot around each of these subjects. So with that, I'm going to end my initial introduction and turn the stage over to Carrie Maxwell, who's a patient advocate for the PKD Foundation and the National Kidney Foundation. You can see her bio below, but the stage is yours, Carrie.
Good morning, everyone. Thanks for being here today. I'm honored to be here today to share my experiences as a granddaughter, a daughter, a patient, and a mother whose life has been impacted by PKD. But first, I want to offer my sincere thanks to the PKD Outcomes Consortium Critical Path Institute for what you're doing to include the patient perspective as you seek to improve public health through including many stakeholder voices in your process. Thank you to the presenters for all the work that you put into sharing your information for not only this conference, but for the work that you do day to day for patients. Thank you to the researchers and regulators who I would imagine work thankless jobs for years awaiting the payoff of their study and work. And thank you to the PKD Foundation and Otsuka, who have a special place in my heart for their work to better the outcomes of people with PKD and their families. Today, I'd like to underscore the generational impact that PKD has on families. When I was just 10 years old, diagnostic imaging confirmed my parents' greatest fear, that I had in fact inherited PKD from my father. Back in 1988, there was very little to be done for someone as young and asymptomatic as I was, so my parents held that news and waited to tell me until I was 18. Now, as kids, my siblings and I watched our father struggle with everything that living with PKD meant. He received home hemodialysis 3 times a week with the assistance of my mother. He received the gift of life in 1987 in the form of a perfect matching kidney. Now, there were extensive follow-up visits, hospitalizations because of reactions to the many medications, including immunosuppressive medication that he needed to take, or close calls with rejection. He had sepsis 5 times and 3 different kinds of cancer. So it's safe to say his condition and continuous need for care were front and center in our lives, and we were acutely aware of how tenuous his health was from moment to moment. So when you get a diagnosis of PKD, it is a lot to process, even when you have no symptoms. I didn't know it then, but my daily life wouldn't be affected by the symptoms of PKD for at least 10 more years. And at that time, we were encouraged not to talk about our disease due to insurance and other discrimination. I'm so thankful for legislation that's happened since that makes this no longer the case. However, there's something frightening that my generation of patients has in common. We're not strangers to PKD. Many of us watched our parents deal with a brutal disease daily, where the only strategy then was to wait until your kidneys failed, then start dialysis or be fortunate enough to get a transplant. Receiving a diagnosis of an unknown illness is scary, but the fear that comes with a diagnosis of something that's already impacted your family life is something else entirely. But through it all, my dad kept a positive attitude about his health challenges. He was respected by his doctors for how well he understood his condition and medications. He spoke up when he felt he had too much or too little of something, and quickly gained the respect of his physicians for his understanding. Through that incredible example of his, I learned the value of being my own best advocate and playing an active role in my care when I can. As an adult, becoming a mother brought the silent progression of my PKD into focus, but it was also a reminder to me that now I had possibly passed PKD on to generation number 4 in our family. I knew that my dad had experienced guilt over this, over passing PKD to all 3 of his kids. And I began to understand this in a more personal way. But I decided that I could channel all of those emotions into something positive and set the best example I could for my girls by taking charge of my health, being open to research, and sharing my story with others. By the time I reached age 30, the silent progression of PKD began to show up in routine blood pressure checks at my general practitioner. And while I wouldn't ever characterize myself as being lucky to have PKD, I am fortunate that I knew from my upbringing what was happening to me at an early stage. I meet so many people who don't get the opportunity to find out in stage 1, 2, or 3 even, that they have a progressive kidney disease and can do something about it. So many don't find out until it's pretty late. So I began seeing a nephrologist at that time and quickly discovered that our visits would be comprised of routine labs, blood pressure checks, and a few suggestions about how I could either lower my phosphorus or raise my vitamin D, whatever the case was that month. I knew quickly that this reactive approach was not going to work for me. A few years later, as part of my self-research, I found AD-PKD trial of Reprise online, the clinical trial for tolvaptan. I took the trial to my nephrologist to get her blessing and began the trial in January of 2016. And I knew within an hour of my first tablet that I had received live medication and not a placebo. It's important to note that not only did I find this trial on my own and not through my nephrologist, as I know now is the case with many patients, but the center where it was being conducted was a 4-hour round trip drive from my home. So over 31 trial visits, that adds up to 5,500 miles and 124 hours of just driving. I say that because I'm blessed that I have an employer that supported my participation in the trial, still supports me as a patient. But I'm fortunate that I've had the means, the energy, and the time to invest in making those trips. But I imagine this isn't the case with all patients. And so as we think about ways we have had to pivot in the last year, I encourage thinking that considers how trials can be more inclusive of patients that can't commit to this kind of time and travel to trial centers. And as we think about inclusion, I commend you for keeping the patient voice front and center as you develop your clinical outcomes assessments. What I want you to know from my perspective is that not all patients are alike, and it's so important to include early disease patients as it is to include those with end-stage renal disease. As kidney patients, we have seen and know all too well what the progression of this disease may be, having watched our parent or other loved one suffer through it. And when we consider the side effects of a breakthrough medication like tolvaptan and how many of us need to adjust our lives to it, we need to be reminded of the trade-offs and consider them in the context of what our disease course may be, if not for tolvaptan or whatever comes next. I'll never be able to find the right words to express what it meant to me to be a part of that trial, not only for the benefit I may have gotten, but for the countless patients waiting for a medication like it, for their parents that hoped and prayed for it for years, and for the next generation coming after us. My dad would have done anything to slow the progression of his PKD if he could have. So for me to have the opportunity to take a medication that does just that while he was still living was a gift 30 years in the making for him and my mother. From the bottom of my heart and on their behalf, thank you. PKD impacts families. I never met my grandmother. She was 44 when she died of a then unknown disease that caused multiple cysts to grow in her kidneys. My father was 43 when he received a kidney transplant that gave us 30 extra years with him. I'm 43 today and in early stage 4, and I have more hope than I ever thought I'd have at this stage in my life. In March of this year, my oldest daughter, Becca, became generation number 4 in our family to receive the PKD diagnosis. As I think about her future and the content that's been shared over the course of the conference, I think about how it will impact her future choices. Today, our choices are participate in research or take the only therapy on the market or do nothing. As she finishes her double major in music and education, I think about what will be available to her to slow her PKD and how it will fit into her life as an elementary music teacher. I think about what will be available to my 8-year-old when she's an adult. As you leave the summit and go back to what you do to take steps forward for PKD, I want you to know that I am one person and family, but there are thousands more stories just like mine out there, each a life waiting on and believing in the hope that research brings to patients. Thank you.
Thank you, Carrie. That was a really great presentation. We really appreciate it. You know, I will tell you one thing. It's really hard to be a moderator after a presentation like that. But thank you. We appreciate you sharing your thoughts and how this disease affects you and your family. So if I can ask the participants for the panel session to go ahead and turn on their camera, that would be great. And we'll go through some quick introductions. So great. So as everybody gets on, Frank, if you'd like to go ahead and give a quick introduction, you can tease me since I teased you about the picture.
Yeah, reality sucks, doesn't it? Anyway, this is me. I'm Frank Serwick. Very nice to see you all and welcome to this very, very important meeting. I really appreciate everyone's active participation, both in the past and hopefully today and in the future.
Great, thanks a lot, Frank. And so for the panelists, Jeff, if you're on, could you give a quick introduction, your title and affiliation?
Sure. My name is Jeff Barrett. I'm Senior Vice President at the Critical Path Institute. I'm heading the Rare Disease Cures Accelerator Initiative. It's something that I took over from Jane Larkindale. This has been quite an interesting transition. It's an important project that requires the integration of a lot of different stakeholders, as you heard during my lecture, but so important. And I really appreciate the journey of the patients here, because the whole purpose for collecting data in the first place is with the intention to benefit patients in a very real and meaningful way. So, and it's only because they share their journey that we're able to do this. So thank you.
Great, thanks, Jeff. Ali?
Yes, good morning, good afternoon. This is Ali Hariri. I am in the clinical development at Sanofi. I'm a nephrologist and look forward to great discussion today. Thank you.
Thanks. I don't think Leslie's been able to join us yet. Hopefully she'll be able to. Are you out there, Leslie?
I am here.
Oh, great. Welcome.
Would you like me to introduce myself? Is that I just got on?
Oh yeah, just please name and affiliation would be perfect.
So I'm Leslie Inker. I'm a nephrologist at Tufts Medical Center in Boston.
Great, thanks, Leslie, and thanks for the talk. That was a great talk. Praveen?
Hi everyone, Praveen Jadhav. I'm a development team lead at Otsuka, leading the gene therapy program. Thanks for having me here.
Thanks, Praveen. Romaldis?
Oh, I'm Roman Machulitis from EMA. I'm responsible for many procedures in nephrology at the European level. I'm a little bit nephrologist as well. I'm involved in many of biomarker qualifications related to renal things. So that's why I think that our meeting today and yesterday is very important to move things forward to meet the needs, which are tremendous, definitely. Thank you for coming and thank you for listening.
Great, thank you, Romaldis. Appreciate that. Everybody knows Carrie very well now. Dorothy?
Yeah, hi, I'm Dorothy Oberdan. I'm the Director for Patient-Centered Outcomes at Otsuka.
Thank you, Dorothy. Klaus?
Hello, everybody. Klaus Romero here, Chief Science Officer for CPATH and longtime supporter of the PKD Consortium, background in clinical pharmacology and epidemiology. And you saw from my talk that I'm a clinician that likes numbers.
Thanks, Klaus. Bex?
I am Bex Beck. I am a clinical outcome assessment scientist within our COA program at CPATH.
Lisa, and we missed an MS on the end of your name.
It's okay, life will go on. Hello, thanks so much for doing this. My name's Eliza Thompson. I am the Deputy Director of the Division of Cardiology and Nephrology at the US FDA.
Thanks, Eliza. Vincente? I know you're there, Vincente. I saw you. Are you on mute?
Okay, sorry for this. Yeah, I'm Vince Torres. I'm an adult nephrologist at the Mayo Clinic with a long-term interest on PKD.
Great. Well, thanks to the panelists. Thanks to the speakers. We have a pretty big panel here, and so I think we're going to get into a couple really key discussions. I'm going to set you guys up and then basically let you at it. I'm gonna let Frank also bring up different points as well as we move through this, but it should be a lot of fun. So about 2 years ago, when we began thinking about for the PKD Outcome Consortium, what were the different opportunities that were available to us in order to be able to progress drug development across AD-PKD and AR-PKD. And so, as you saw how we set up the talk, there were 3 basic themes that came out, right? Biomarkers, including TKV modeling, COA measurements, and then data sharing. And what I'd like to do within this panel session is basically work through those different topics. And so I think the very first question that I'd love to talk to the panel about is, what are the tools that we actually need? What are the most important tools to go ahead and approach these unmet needs? Of course, with the leading tagline that it has to be something that matters to the patient, right? That helps us be able to create these therapies for the patients. And so, a couple things that were talked about yesterday that I was able to pull into this session was, moving away from functional endpoints like GFR to more direct measures of tissue injury. Should we be concentrating on biomarkers for AR and AD-PKD, or should we just concentrate on one of those diseases, right? I think we know the answer to that. We should do both. Should we be concentrating on not only renal biomarkers, but then also extrarenal biomarkers? And then what about biomarkers for the pediatric population, not only in AR, but also in AD-PKD? And then further development of imaging biomarkers. beyond total kidney volume, very, very much where Vincente's talk went, right? How can we expand that? And are there other imaging approaches that we should be using? So I'm going to initially open this up to the speakers, both Vincente as well as Klaus, to get some of your comments. But Vincente, if you'd like to go first, and then we can move to Klaus.
Okay. Well, I think cystic expansion, kidney cystic expansion, represents about 80 to 90% of the disease. So it is the disease. And therefore, addressing that is, I think, is very important. And I think talking about the patient experience with the disease consists mostly of pain, either due to cyst bleeding or stones or infections, all of which are related to cystic expansion. It can be just organomegaly, which is also cystic expansion. Kidney failure, which in most cases is related to cystic expansion. And then the anguish that they experience because they are anticipating, based on the family history, the development of kidney failure and the development of pain. So this is the way the pain, and we have heard very well this morning already, is the way the patients experience the disease. So cystic expansion in the kidney is very important, and because of that, the CRISP study was initiated to see whether actually total kidney volume and kidney growth was associated with these outcomes, particularly with kidney failure. And that was demonstrated by the CRISP study. And this was has been expanded and confirmed by the PKD Outcome Consortium, which has led to the approval of TKV as a prognostic indicator. And I think at the present time, this is the main and the most accurate prognostic indicator that we have. However, I think total kidney volume does not completely capture all the angles of cystic expansion. And then there is significant room for improvement. When we developed the Mayo classification, was trying to alleviate some issues about atypical cystic expansion that are not associated with the development of kidney failure. And so in that way, the Mayo image classification is a small improvement on just using total kidney volume or height-adjusted total kidney volume. But as a prognostic indicator for selecting patients for clinical trials, this is not really very important because in the atypical cases where the total kidney volume does not predict decline of kidney function are only maybe 5 to at the most 10% of patients with polycystic kidney disease. However, this is very important for the individual patient. So, which is not what we are talking here today, but for the individual patient, when the clinician meets the patient and has to give an individualized prognosis, you cannot rely just on the total kidney volume without looking at the images of the kidneys on a good MRI or a good CT scan with contrast. And the reason because you cannot do that is because you cannot exclude these atypical cases. So there are, in the images, there is a lot of information that is not used right now. One feature that has not been used is the cyst number. And so very easy to understand that 3 cysts of 300 mL cause much less damage than 900 cysts of 1 mL. And that's because really where the action occurs is at the cyst parenchyma interface. And this action at the cyst parenchyma interface occurs by several mechanisms. One is by the release of cytokines and chemokines that induce inflammation and interstitial disease. And the other one is just because of mechanical pressure. So you have this other feature of polycystic kidney disease that causes kidney damage. And this can be indirectly measured by cyst number or measured in the cyst parenchyma interface. But there are other ways that you can do nowadays. You can actually segment the cyst and the non-cystic tissue and separate them together with modern imaging processing and analysis technologies. And then you can take the non-cystic tissue and do some analysis on the non-cystic tissue using texture analysis or MRI fingerprinting or other technologies. So there is significant room for improvement. And that's something that unfortunately is not at the present time available to be implemented in clinical trials, but it should be in the future and improve the prognostic value of these imaging biomarkers and maybe make them more attractive to be used as a surrogate biomarker.
Great, thanks, Vincente. I know that I've seen also that there's been a lot of artificial intelligence approaches to doing image analysis as well, and that probably should be a direction that we explore. But let me turn it over to you, Klaus. You know, what are your thoughts, right? Even go beyond, you know, total kidney volume in the direction that we could go as a consortium.
Yeah, no, and thank you, Vicente, for that great setup. Because essentially, I mean, it's capturing that first bullet there. It's all about understanding how the individuals in their journey with PKD feel, function, and survive. And so, but then how to measure that? How do you demonstrate that you're actually impacting positively how patients living with the disease feel, function, and survive? You need the clinical trials. And the question then becomes, how do you design those trials with measures that actually capture how the patient feels, functions, and survives? We've made tremendous advancements thanks to the consortium, the experts like Vicente, the industry collaborators like Frank and Pravin and Ali, and of course, you guys, the patients, Gary and the PKD Foundation representation. We have a surrogate. That's fantastic, but we still need to understand how to best design optimal trials with that surrogate. And what Vicente was mentioning is essentially about really understanding the sources of variability, how you can tell those subpopulations apart in a really, really accurate way. And if that means that you also need to think about different types of measures that may go along with volumetric measures and functional measures and even patient-reported outcome metrics, that's when you'll have a truly powerful setup to develop an ecosystem that levels the playing field for everybody and makes PKD a truly interesting area for investment for sponsors in the pharmaceutical industry. And who's going to benefit at the end of the day? The patients. And that's what the consortium is about. You mentioned artificial intelligence, Joe Michael. Of course, that's fantastic. The more we can truly accurately measure the volume and the volumetric changes and the evolution of those cysts, but that's, if you think about it, that's empirical, right? It's just measuring how the cysts are dynamically changing. Vicente mentioned something really important, the mechanistic aspect. You can take that mechanistic aspect and put it in the preclinical stage and also improve the translatability. And that carries a tremendous amount of value for industry so that they can properly advance or not advance products that actually carry promise and not advance products that may not carry promise. And so that's what I see as the consortium focusing on for the future, to continue to move the needle as the consortium has significantly for medical product development in PKD. Stop with that.
Great.
Can I
Thanks a
ask?
lot, Klaus. Oh, please.
Yeah, I wanted to ask a question, part comment, part question to the panelists about some of the things that both Klaus and Vicente mentioned, and that has to do with mechanism. If we're talking about biomarkers, TKV or a biochemical or some other biomarker, the idea is, and what I'm inferring from what Vicente said, is that some of these things might be on the causal path for kidney destruction or kidney function, which is, ultimately that outcome that Klaus is for us to be able to improve. What does the panel think about the combination of TKV with inflammatory biomarkers or other types of data that could be acquired through the various omics efforts that may be ongoing in the area of PKD, to sort of create a more granular score, so that in your example, Vicente, if you're comparing two people with very similar kidney profiles, they look like they have the same volume, the same general number of cysts or distribution of cysts, but they still end up with different renal function. Why is that? Is it one's more prone to the inflammatory or fibrotic aspects of the disease, et cetera? And can we measure those? Can we measure the things on the causal path that matter, and then use those biomarkers as readouts for a drug's efficacy based on a particular mechanism?
Well, I will go first. Well, I think we can do that. The problem with this is that we really don't understand enough mechanistically what these biomarkers will be. Like in what I would say about the cyst parenchyma area and inducing an inflammatory reaction, yeah, that's fine. But the capacity of different people to respond in an inflammatory response will be changed from one patient to the next, because we all react differently to it in inflammatory responses. So I think understanding what these biomarkers are, a biomarker that is very attractive, which is the one that Dalila McCallier spoke about yesterday, is MCP1. And the reason why MCP1 is so attractive is because first, Dalila found that in children, at very early stage of the disease is already increased, but there are some animal studies that show that when you knock out one of the PKD1 genes, you have increased expression in the tubular epithelium of NCP1 before there is any macrophage around, and macrophages appear later on. And there is some studies that have shown that actually there is a direct link between PKD1 cilia and expression of MCP1. So it may be so intimately connected mechanistically to polycystic kidney disease that makes it a very, very attractive biomarker. So I think other biomarkers may be much more distant, right? The kidney injury biomarkers, generally inflammatory biomarkers, they may be good, but may be quite distant and not very specific, because they can be abnormal in many, many other situations. So that's, I think, my comment about other biomarkers. I think finding biomarkers that provide added value to the total kidney volume would be fantastic, but that will need to be proven.
Yeah, Frank, if I may add, I mean, this goes to the holy grail, even what Carrie talked about, in terms of, at the end of the day, these biomarkers have to be predictive towards the progression of EGFR. I mean, we even heard from her that slowing the disease, even to the extent of reversal of disease, is very important from patient family perspective. And then not only that, I think we'll have to show that drugs actually can reduce that level. So there is a treatment-induced change, because that's where the second predictive power will come. Not just that that initially this biomarker increase, but we can actually make a meaningful change. And it was again going to require data sharing, or even to the extent of going back to existing data, existing samples, reanalyze them and say what did we see, because I think a lot of these biomarkers were probably unknown when we did the tempo or the reprise and other studies. So I think this will also require us a little bit of collaboration to go back and look at the samples and say how has not only the biomarker had changed at baseline, but has treatment actually meaningfully changed that, because that's where the translation to treatment would come.
Now, Pravin, I want to follow up on the point you made, because that connects well with actually a couple of questions that came in from the audience via the chat. The notion of additional biomarkers or measures of any kind needs to be seen through the lens of how actionable are they to meaningfully advance drug development. In this conversation we're having today, the daily patient care, that's an important conversation, but that's a different category of discussion. You mentioned something that you being a quantitative person, and Jeff and Ali will also resonate with this, the learn and confirm paradigm. You start generating some data and some knowledge, you confirm that, you put that into the context of the big picture, you leverage the data, then you confirm again, and then you learn more. And so there's a question about measuring functional kidney tissue. That sounds exciting. Question is, do we have the data? Do we understand how to put that into context with something that is actionable for drug development, either to understand the pharmacodynamics of a drug or to understand the dynamic changes of the disease, or both, and how to put that in the context of clinical trials. And then Ron, as always, made a great point about modifying genes and the impact on biomarkers. That's an absolutely valid point about the sources of variability. You need to understand what makes a biomarker tick and how to distinguish different subpopulations in terms of their dynamic behavior. Just wanted to address those couple points.
So thanks, Klaus. I think that was really good. What I'd really like to do is I'd like to now pull in Elisa and Romaldis to get their feedback around this conversation and where we're going. I am really excited by that question that was in the chat box. So please put more questions in the the Q&A box. Please put more questions in the Q&A box, audience, and we'll try to address them in a seamless manner like Klaus just did there. But can I call on you, Elisa, to get your opinion around this conversation and where we're going?
Yeah, no, thanks so much. And it's been fun to listen to the discussion thus far. It's also been amazing to see how far this community has come, I think, in the last 10, 15 years from where we started to where we are now. And I just want to make a general comment, and I think it ties to some of what was discussed yesterday. You know, ultimately, a trial is a tool, right? It's a tool to effectively and efficiently figure out if a drug is effective, right? Or, you know, does what you hope it does, and to characterize safety, and really to get sponsors and to attract sponsors to a space, you have to be able to make the business case that you can do effective and efficient trials, right? Because they could develop drugs for other diseases. You know, I struggle a little bit with this discussion that we're having because it's just about sort of what biomarkers do we want. But I think the issue is for what purpose? When I look at the PKD space, which is perhaps the one I'm more familiar with than autosomal recessive disease, you know, I think we, you know, and what I hear from the discussion as well, you know, there are a number of unmet needs here. We need better tools for predicting and you know, sort of forecasting, you know, who is at high risk of progression. And this is a tool that's valuable for a clinical trial in terms of who you enroll in the trial. It's also valuable for clinical practice in terms of clinicians figuring out who they need to treat. We refer to the disease as ADPKD, but as we're learning, there are probably important subpopulations here, and that we really need better tools to say this is the population where maybe it's more the, you know, if it's a fibrotic process or something's different here, right? We need to be able to segment the population, so we need those tools. And then finally, it's absolutely true, to do this effectively and efficiently, we need better response biomarkers. And that's both response biomarkers that can support our decisions about proof of concept or selecting the right dose in phase 2. You know, they give us an early, quick readout. It's not the perfect readout, but it gives us the signal, but then also need to refine our efficacy endpoints. And the last comment I want to make is, please don't throw out the baby with the bathwater. I understand that, you know, EGFR is not perfect. I understand that TKD is not perfect, but hopefully what you heard from both Vincente's story yesterday, as well as, or not yesterday, but the videos in Leslie, is that you can continue to refine the endpoints you have now to do a better job. It doesn't mean you should look to new ones, but a lot of work, important work, has gone in, and I think they do provide a path forward, at least for the time being. So thank you.
Thanks, Elisa, for your important comments. Romaldis, would you like to comment?
Yes, I completely agree with Elisa, and regarding the last comment, don't throw the baby I think that what we discussed yesterday was that GFR is not so perfect in the early phases when you have a hyperfiltration, when you have a not so informative GFR measurement. And TKV here is a definitely advanced tool for the understanding of how progress is going. And of course, what we also mentioned that despite that, I think that at least in my country, in my area, in other part of the ocean, we would never approve based on TKV only if there would be no supportive data by the GFR. And if you can see that what made the tolvaptan life is not because we had a survival data which was very, very usual, but because we had a strong support by kidney function showing that no worsening is there. So what we need to show and address the issue of the patients that okay, not to be too late. I think that to catch the progression as early as possible, and here as a regulator, I am want to say something as a scientific regulator, want to say another thing like that monkey which does not know whether she's beautiful or she's clever. I think that maybe I am too scientific here, but I think that okay, generally we should understand that biomarkers for the exploratory purposes, for the first early phases, this is one story. And for the trials to understand how the potent byproduct is. Totally different goal for the biomarker is for the confirmatory study. So and here we go to more traditional, more early, and I think that we are talking about all of them. So for the early markers, and I speak about the scientists, I think that most probably I am the only one in this room of 131 who is believing in the better characterization of the hyperfiltration, which is a not a PKD issue. It's an issue of the much broader nephrological area, let's say diabetes as well. And I think that our colleagues from the MRI area might see that yes, we can do more with this functional MRA. Like you remember, we saw even mentality working through the MRA type of things, maybe or maybe not. And I fully agree that to strengthen TKV with the inflammatory or epithelial damage or fibrosis aspects. I saw in the Q&A section also the functional kidney tissue. Yes, this is also very important marker saying where we are at certain stage. So to support somehow, it would be really very important for the early, of course, early clinical trials, not for the later. So I think that that's it. What I think very important to consider as well. Thank you very much.
Thanks, Romaldis. I appreciate your comments around this, and I think what we're finding, more and more, I think we can reach alignment around this, how we best go forward with bringing these different biomarkers forward, and in a way it doesn't endanger, a given development program, but yet we can learn from that. So I think that's extremely important. There is one interesting question here, and that's to the regulators. So before we leave Lisa and Romaldis on this subject, would a regulatory agency approve a drug only if it improves renal function and not total kidney volume? I think that's interesting. I think your answer can really enlighten how you're thinking about total kidney volume and kidney function. So would either of you like to address that question?
I'm happy to address it, and I'm sure Romaldis is as well. So, I think the question is, again, these are tools. And so what are we trying to understand, or what do we think that the impact on GFR is going to predict? And I think, we've heard from patients, and understandably, that one of the things they want to avoid is progression to kidney failure. And so to the extent that we think that what we're seeing in the trial means that we will have a meaningful impact on slowing the rate of progression of the loss of renal function, such that will delay the onset of kidney failure, very much so. I would note that some therapies may not affect the size of the cyst. And would even note that with tolvaptan, it has, a lot of the effect, and someone from Otsuka should correct me, but I would say there's a large pharmacodynamic effect on the cyst. By that, I mean it reverses when you come off therapy, but yet the trials showed also with compelling data that it was slowing in a meaningful way progression and progressive loss of the kidney function. So it may be true you're not going to help patients with the symptoms associated with large kidneys, but certainly I think that we would approve a product and certainly sketch out for whoever used it in the label what benefits you were getting, and make it clear that what it was affecting was the rate of loss of renal function. Hopefully that answers your question.
Thanks, Elisa. Any comments from Aldous before we move on?
Most probably not. Thank you. We'll move further because regulatory is always very traditional. You know that even GFR is now sort of the novelty, although not finished for the as a surrogate marker, as a slope, as a certain cutoff. You remember, we will come to that later, that these are more or less clear surrogate in some cases for more traditional markers, which were always what we relied before. So, and to skip or to switch from GFR to something else in addition to PKD, it would be very difficult. At least for the confirmatory studies.
Thanks for all this. What I'd like to do is I'd like to really move us on to the second point, right? And that's around clinical outcome assessments. And so, Dorothy, if I can call on you in a moment, but I think your presentation did a really nice job in defining an area that we can concentrate on, and that's around pain and discomfort associated with the disease. I think one of the questions is, and I know you did a very nice analysis within your presentation, are there other areas that we should look at as well? Not only in AD, but then also in AR PKD. And I don't know what analysis you've done there since your talk really concentrated on AD, but I'd love for you to comment around that.
Yeah, so I think what we did with that analysis that you're referring to is that we looked at the information that we collected with various PRO instruments across, I think that was from the Overture study. So it was a non-interventional study with over 3,000 patients, and looked at those patients that had good outcomes on the PRO measures versus relatively bad outcomes on the PRO measures, and see where those patients landed with the traditional PKD outcomes that we kept collecting throughout our development program. So the one thing that really stood out was that kidney pain was by far the most impacted on the quality of life perspective. And so we haven't specifically teased out why that is, because in that particular population, we didn't do very intensive TKV monitoring, for example, and we didn't do very intensive eGFR monitoring. So we have very limited data on that. But it certainly is what we also know from the development process when we did that AD-PKD PDS scale, where we talked to several hundred patients there. It really kind of is a pain is a very complex experience for PKD patients. So it really impacts quality of life on several levels. It's related to the kidney size, the growing kidney that came up as themes. But there's also other areas where patients weren't really able to identify exactly why they were experiencing the pain, and we couldn't find an explanation of why that is. It could be related to the blood pressure. Maybe there's some blood pressure spikes that cause more headaches or other types of pain in the body. So that is kind of the thing. We need to do more research to find out why that is, where that is coming from. It's not just related to the kidney.
Great. Thanks, Dorothy. Bex, can I pull you into this conversation? I think you gave a really nice talk on how, we could work through, enhancing the current scale and overcoming some of the deficits that Dorothy has brought up. But can you give us your opinion on, not only the pain scale, but are there other opportunities out there?
Yeah, I think there's most definitely other opportunities out there. Developing a measure and going through this process is very iterative. And if it was easy, everybody would do it and we'd already have an answer, right? And if you've talked to one patient, you've talked to one patient. And others have emphasized the importance of what is important to the patients, and that's essentially what needs to be done, and where we probably need to go back to is to learn more from them and their experiences. Dorothy's already done a great amount of work in this area, so learning more from her experience and building on the work she's done and emphasizing that collaboration, I think, is going to be very important.
Great. Thanks, Bex. Going to my industry colleagues, Frank, Ali, Praveen, what are your views on, the value of pulling in additional COA measures into clinical trials? Do you see that as a major gap right now, something that we need to fill as drug developers?
Can I just comment with regard to some of the things that Dorothy shared?
Absolutely.
It's important, I think, as we do drug development, and there have been a number of initiatives, FDA, NKF, and others around these things, to understand what the disease's impact is, and then what the drug's impact is on the disease, or what the drug's impact is on the way the patient perceives their disease or perceives life in general. So I think it's always a good idea to ask the patients how they're doing in a trial beyond just what AEs are collected or things like that. The thing that was important to me, I think, is that Dorothy mentioned the Overture trial, which was not a interventional trial. And because it wasn't an interventional trial, it was able to focus on a broad population of patients that wouldn't be studied in a drug trial. Pediatric patients, patients that have already gone through transplant or were in dialysis, to really understand what their impressions of the disease were and how, for example, pain varied over the course of the natural history. That is important for everyone to collect. And here I'm talking about academics. I'm talking about I can't pronounce the acronym, but the PKD Foundation's registry and NORD's registry. That baseline of data is really important. But then you layer on top of that, what can a drug do? What can a drug do to improve quality of life in general and performance and all of that? And then what do you give up when you're doing that? Are there issues with the drug that could be more limiting, or that address some aspects but maybe worse than others? That's where I think PROs really should be kind of a standard across all trials, interventional or non-interventional.
Thanks, Frank.
Yeah,
Praveen?
I would agree with that. And I think, and it goes to some of the previous discussion that, focused on disease, TKV, EGFR is important. We are trying to improve upon the disease aspect of it. But at the end of the day, it's also about how patient feels, how they perceive the progression is affecting them, how they perceive the change in progression is affecting them. is also important, and also how, I mean, the risk benefit is at the front and center of this, because these drugs also come with side effects. How the side effects actually are either worsening that benefit that may be coming along with it. And that has to be measured. And I agree with Frank, both interventional and non-interventional trial, and even for some of our studies, and Frank will remember this, he created a questionnaire, part of our phase 3. Would you take this drug for rest of your life? I mean, that's actually very important to know from patient perspective. I mean, that those are some simple questions and may not get to a validated PRO level, but we got to constantly ask those questions. And I think what I would say is, from collecting is one thing, but then translating into either a label or even for the payers, this is what this PRO or this measurement or this delta that we have achieved in a trial means to patient, because that will not only allow us to translate what we are delivering beyond just the functional benefit, but also the holistic disease benefit towards it.
Ali, I don't know if you have a point to make here, but you're welcome to join in.
I just wanted, this is Ali Hariri, I just wanted to actually repeat everything everybody else said, but just recently in another disease, we had the same experience. I mean, it was Fabry, and frankly, it took us more than 12 years to be able to confidently have a reasonable PRO. So interview with the caregiver, physician, patient, moving to larger scale interviews, moving to observational studies, and then to small interventional studies. And again, we now finally feel comfortable that we have something that allows us to actually capture the changes over time in the patient, and also what is a meaningful change that is important to the patient. So all of these things, again, I think, frankly, the critical path has been pioneer in this. I'm bringing coal to the, anyway, I don't think I need to emphasize it anymore. Thank you.
Thanks, Ali. So I think what I'd like to do is I'd like to hear from Romalis and Eliza. You know, are these tools that we're missing in our toolbox, right? Is this something that you feel, as you know, reviewers of the applications for drug authorization, that's needed and we should concentrate on?
I may start briefly. It is definitely important to objectively assess how patient feels, functions from his point of view. And let's say, coming back to the same 12-up-10 authorizations some years ago, we also made this quite clearly that key supportive measurements was the worsening of or let's say exaggerated kidney pain, which we named as a medically significant, and we defined that as a requiring prescribed relief, last resort analgesic, narcotic, or nociceptive, you know, which is sort of clinically meaningful event, and this is something which always works. How far we can go in the structured manner, not leading to case by case for this or that product, is another question. And here we go to this all broad PRO scales. We had lots of guidelines how to develop them, and I was surprised that it took 12 years to the Fabry. But I can imagine with the limited number of patients, it should take a time. So, but this would help if you will have the PRO nicely describing the minimal important difference. Can you base statistics? This difference is based on, let's say, more than half of the patient, of course, because not everyone is similarly sensitive to something, or to the same pain, or to functional burden. then this would definitely be supportive. Could it be a basis for the approval? Well, I'm not aware of any approval based on that only. This would be a very strong supportive biomarker. The endpoint mitigating, let's say, lack of other data like GFR or something else. And I personally really believe, and that's why I think that it's very good that the patients in this section, as in previous, they clearly express their feeling of the disease. This is less than hypertension, that you don't feel the hypertension. Here you feel the disease. So now we as a regulator, as a scientist, we need to put these feelings in the numbers. How to do this? Yes, of course, patient perception is one. Supported with the observers, with the professionals' observation or rating, and this would definitely work. And I'm happy that the things were done already. I think that now we need to put them somehow into the like a master tool for the others, and test in one and another product, and it should fly. Yes.
Thanks, Romualdus. Any comments, Eliza?
Yeah, and I'm hoping we'll also end with input from Carrie on this one.
Yeah, absolutely.
I think the concept here is that drug development is supposed to be focused on the patient, and we need to understand what matters to patients. And as we're hearing, and I think we've heard this before, but maybe we're hearing it more loudly, or maybe just because of the wonderful work that Dorothy has done and shared, that pain is an important component of this disease. I think the challenge here is that the pain has many, perhaps different etiologies. It can manifest in different ways, and so that's a challenge. But doesn't mean we shouldn't try to better understand how to study the pain and find tools that can assess the impact of therapies on pain, with the understanding that some probably won't impact the pain, but others via some mechanism might. And I would think, that if the therapy were to show that it, in addition to doing something else, actually impacted a patient's pain, my goodness, the patient should know it. We should put it in the label. We should describe it. It's an important benefit. But I just want to sort of say, before turning it over to Carrie, is I do want to thank Dorothy for sharing her work, and Suka, I guess, for sharing it, because I think that this is the key. What you've done thus far, it has moved the field forward, I think, in terms of our understanding. But obviously others need to step up to the plate and do more and contribute as well. So thank you for your willingness to share and for all the work you've done.
Thanks, Eliza. And the nice part is you addressed a couple of the questions that were actually in the chat or in the Q&A section around the fact that we need to understand where this pain's coming from, et cetera, and the mechanism behind it. So thanks for doing that. Carrie, I'd like to pull you into this conversation, because I think this is very germane to your presentation you gave earlier. What are you thinking about this this discussion?
Well, obviously not a researcher or a scientist or a clinician, so I'm trying to hang with you. Just the topic we're talking about most recently, and it is something that even patients in the chat were asking me about. I'm fortunate I can't relate to the pain scale as far as my kidneys are concerned. I've had 3 children, so I understand pain. Maybe it's in the context of that when I'm asked about my pain, I have it all kind of that I'm thinking about, like, well, I've had 2 children without drugs. So just to be plain, since we're all friends. What I will say is there was a term given to me in the clinical trial that unlocked this idea for me. It was called awareness of your kidneys. It's more, I'm aware that they're large. It may not be something I can say to you is I'm hurting and I need to take something for it. Because we probably, err on the side of not taking medications because we know what they do to us over time. And, if it's something that Tylenol or something like that doesn't help, well, we can't take an NSAID. So, we're not going to do that. We're just going to use other methods, heating pads, et cetera. But what I will say is just that term of awareness of the size was something that really, really changed in my mind about, okay, now I have something I can say. I'm aware that they're causing me to have to buy high-waisted pants, things like that, wear more dresses, like things that you maybe don't talk about in a clinical setting, but that impact day-to-day life, because of what this does to your waist, what this does to your waistline. But on a different topic and something that maybe isn't talked about a lot, and as we talk about taking two pills a day if you're taking tolvaptan, or if we're talking about other therapies that come down the road, something I didn't point out before in my talk, but I think is maybe something to consider, is the mental and emotional aspect that this disease has on people, even people that don't have symptoms. So, for instance, I would consider myself someone that, besides blood pressure, I don't have a lot of symptoms that I'm treating, but I'm on a medication that I take twice a day because symptoms are coming for me. So it's I think something that we talk about, that we understand. We go from our disease being rather silent and something that we're not focused on until it really does present and we have to start dealing with it, to something that now we're taking something twice a day and we're reminding ourselves twice a day, we have this disease, we have this thing that's happening to us. And on top of that, we don't know what's going on inside of our body. We only can go every 3 months and get our lab work done and hold our breath for the next 48 hours while we wait to see what happened to our EGFR or our creatinine or other values that are important. So there's something about just the emotional aspect of it that I feel like I just want to share is a thing among patients, and I'm sure that's not a surprise or new information to any of you, but I just wanted to make sure it was commented on because it's a real thing, and I think that impacts people's lives just as much. Maybe that's something they're not able to articulate, in a clinical setting, because it feels like we go to the doctor for aches and pains, not to say how much I'm worried or stressed about what this disease is actually doing to me as well, what it's doing to my ability to earn money, what it's doing to my ability to bend over and tie my shoes, like things like that.
Thanks, Carrie. I think that was really important to understand those aspects, so we really appreciate it. We are running a little bit short on time. I know there's a couple of questions in the chat, in the Q&A box. We'll get to those post meeting most likely. But thank you for those questions. What I'd like to do is I'd like to move on to data sharing and really pull Leslie into this conversation. I think she gave a fabulous presentation on what she's doing with chronic kidney disease and change in EGFR. So Leslie, can you give us some of the highlights from your presentation about how you've done successful data sharing and really the importance of data sharing?
Well, I'll start with the latter, the importance. I really think that our nephrology community is so generous to each other. I'm involved in 2 different collaborations, one on the endpoints and one on GFR, and more a little bit further away on prognosis. And just in general, the nephrology community is extremely generous. We're very collaborative, and we've made tremendous gains because of that. So thank you to everybody, if anybody's on the call who's given me data, and Vicente has, and we've had lots of emails over many years. So really very grateful to the collaborators. And on one hand, and I've been involved in this for 18 years, and so I think the second word I would use to describe how one of the ways that we are successful, I think, is relationships and value and respect for all the work that's been done in developing the data that are shared. And you know, every email is a different relationship that I'm writing to update the potential collaborator or to ask clarifications on the data. And you know, my research coordinators over many years will know that I look at every email and I really think about who's at the other end and what exactly my question and where are they at to understand where they are. The third thing I would say is that it's much, strangely enough, much harder now. In some ways, it should be easier. There's a lot more understanding in the general medical community on data in the public domain, and that we should all learn from each other. But it's actually added a lot of barriers. And appropriately, we should be concerned about the participant safety and make sure they're anonymous. They participate in these research studies, not so they could be known. Absolutely. But there's seems to be in some ways a lot more barriers. So now it's in some ways it's harder to get data than we used to. And sometimes the development of the data use agreements can take years. Definitely a year. We've been working on one since last June right now. And they'll send us these data use agreements, which are unrelated to the work that's being done. And so we have to shed them away because we have multiple institutions who we're working with to do the work and do the statistics. So we have to get everybody's approval, all oral lawyers at each institution and lawyers at the sponsors end. And I'm telling you, I have to say, I'm not sure that we're doing any good for protecting the anonymity of the patients with all of this. So we're trying to make our way forward in some of those challenges. And maybe I'll end with one more point, is that once we get the data, we really need an intense amount of organization to make sure that we're speaking the same language amongst the data sets. And kidney failure, a simple definition, is treated differently in different studies. The International Society of Nephrology had a conference on definition of kidney failure just before the pandemic started, and I hope that helps because the first thing we'll do is say, well, how is kidney failure defined? And, oh, dialysis wasn't included. Okay, can we go back and just make sure, can we actually get the dialysis inclusion or, you know, various different things? So it's, you know, to summarize, perseverance, relationships, respect, and really I'll end with how I began, with gratitude.
Thanks, Leslie. I think that's really helpful to understand. And you know, congratulations. You've been extremely successful. And I think the reason why you have is you said it the right way. It's relationships and trust. So thank you. I'm going to call on Jeff, but before I do that, Klaus, you wanted to make a quick comment.
Yeah, I'm just going to pass it on to Jeff, but real quick, the point you made, Leslie, is about finding the good middle ground between the push for open science and data sharing and the all important push for patient privacy. Jeff is going to comment on how we've been able to accelerate that and actually get a mechanism in place to integrate data in a standardized way, number one, so that you can make sense of multiple data sets, but also doing it in a timely manner. So Jeff, go for it.
Thanks. Well, before I go for that, Cal, I have to reach out to Leslie and say we should have a beer sometime, because I have the same frustration sometimes. You know, we'd like to create this culture of sharing, but the reality is there's a lot of different opinions on how to do this. And I think, again, to Klaus's point, the details matter, and to be able to convince people that having a best practice approach that includes common language on data use agreements and understanding of the value of the information. You know, there's all kind of opinions about the diversity of data that goes in here and the necessity to integrate this so that we achieve the purpose. But I think there's sometimes over lobbying for this, and then it just prohibits things. But to the point that Klaus is making, one of the things that we spent a lot of time on is putting together a system that has a lot of complicated heuristics. So some of this checking happens matter-of-factly. We work with sponsors on standardizing some of the languages. We ensure that the duplications are addressed in the data. So the details on making sure the data is well curated, standardized, and available to achieve its purpose are happening coincidentally, so that, some of those limitations that could be problematic are avoided at the get-go. But it's a lot of tedious work, but that's the work that has to be appreciated from the standpoint of the value. But nothing happens until the dialogue. And I have the same experience where you're sitting on the phone with a parent, with a patient, and they ask very detailed questions about what their data is going to be used for, how it's going to be used, the issue of privacy. So we've tried to create a lot of fact sheets, a lot of video content explaining this, making sure that we're reaching in each individual audience, because they have different perspectives. They all have, I think, a position that needs to be listened to, heard, and recognized as we move forward. We're trying to create profile views so that each stakeholder's experience is customized to that stakeholder. This involves, again, more work in terms of profiling them and making sure that their experience is optimized. But that's the dirty work that has to happen, coincidental to getting actual shared data to use.
Thanks, Jeff. I mean, one of the biggest data sharing champions that I know is Eliza. Eliza, do you have any comments around data sharing and how that, can help shape, our future as a consortium, as a community?
Yeah, I think, again, when I think back to when I joined FDA to where we are now in the kidney space, we were in a setting where trials weren't being done because we didn't really, I think the sense from industry was it was too hard to do them. I'm going to go, develop my drug for oncology or for another rare disease that is in this space. So I think that the work that's been done by, your foundation, I think the work that Leslie and her group has done has made the renal space what it is today, which is essentially, I think, it's an incredibly exciting time in drug development. Industry is interested. We're seeing trials done. So yeah, it's what's needed to find effective therapies for for patients and people living with kidney disease. It's without data sharing, we just will never get to where we need to go.
Thanks, Eliza. I know, Frank, this is one of your passions. Do you want to make a couple comments as we slowly run out of time here?
Yeah, I was actually hoping you would. One of the things that happened with PKDOC, first of all, I agree with everything everyone has said so far, and I'm very grateful that PKD Consortium took the time to develop the CDISC standards for PKD, but I don't necessarily think that that's enough. I think that there needs to be other levels of collaboration and of standardization that makes it easy for everyone. So I'd like to ask Jeff this. I'd like to ask the folks at the PKD Foundation who've started their own registry, and certainly Leslie, would it be helpful? Would one of the things that the Outcomes Consortium should be thinking about, would it be helpful to start developing standardized CRFs or standardized ways of capturing data that is essential and agreed upon as essential for all trials, whether interventional or not, for this disease? And by making those publicly available and sharing them, saying, this is a core, you should always include these, then there are other optional ones perhaps. Whether that might be helpful in facilitating how industry is data, how academia and these various registries are collecting data.
Not only is that a great idea, I think it also gives the patients early guidance who are participating in those trials that their data is going to be used for sharing purposes. So I think from a transparency standpoint, as well as an opportunity to motivate them in terms of the utility of their data, it's a great idea. Plus the focus on standardizations can only help. Agree completely.
And to that point, I mean, those CRFs are something you can answer, with very little information. MRIs, lab data, things like that may be more challenging. I think lab data, in particular GFR or creatinine measurements, might be something that electronic health records could help with. But kidney volume, really expensive to collect, really challenging to standardize and interpret. I'm wondering whether, another effort to make it easier to collect it. Sometimes industry or people won't collect it because there's not a quote unquote need. But for PKD, I sort of think and agree a little bit with Carrie that not knowing where you are in your disease is kind of like driving a car without a gas gauge or without a, I guess, without a battery gauge in terms of where you are nowadays. And it should become a standard of care for all patients with PKD, and then something that would be added to the electronic health record in a fashion that would be interpretable by some of these registries and consortia, so that it can be translated to useful, actionable data and added to the various models that are being developed to help predict whether a patient is on a rapid or less rapid trajectory and whether a treatment is actually benefiting them or not. So, I mean, that's my wish list, I guess, for this topic. But I'm hoping that maybe the PKDOC and others can work towards that.
Any other comments from the panel? We have a couple minutes left, and then we're going to close this session.
It's about, if I may briefly, I was also always surprised why the companies are collaborating with the data for general things, not for the particular product, but for general things. Until I understood that it seems that some of companies can benefit from that as well, when they can test what they have somewhere close to garbage for something else, and they can test and see the tool, which is everywhere in the publications, in the science. Although, you know, when we speak in the biomarker qualification procedure, whatever, you see that it will not fly, and of course you might see the need not to test or to test something else. So these internal things which are outside of the company, they are always good to know as early as possible. And I think that you should, now I'm asking the companies be more active, you know, in delivering, in creating and supporting all these quite general, quite horizontal discussions, because this is for benefit not only of the patient, but also for some other company, for the particular companies and for the products which are almost, as I said, close to garbage as well. And just a small remark.
Yeah, thanks for that call to action, Romaldis. I think that's absolutely right. We have to figure out how we can share this data, right? We know we can't share proprietary data, nor do we want to, but I think what we need to do is be able to share the data to help drive towards these tools that we're trying to develop and qualify. So I'm gonna close this session. I'm sorry if other panelists had things to say, but you know, we'll definitely get that in the manuscripts that we're going to be writing from this summit. But you know, number one, thank you to the speakers. I mean, great presentations. Audience, if you have not seen these presentations, I really recommend them. To the panelists, thanks a lot. Great discussion today. You were a big panel. I'm glad everybody was able to keep their comments highly focused, because we got through a lot. And so this discussion is really going to help us understand where we want to take the consortium next, who we need to collaborate with and why, and how we need to share data. And so again, you know, I love the CDIS standard conversation at the end. I think that's really important, and how we can standardize some of our data collection. You know, it turns out that we are working with some of the PKD centers to look at how their REDCap databases can be standardized with similar STTM or CDIS language. So that's really exciting, and I think really important. That will help drive things and standardize the approaches. I think the other thing is, you know, talking about the biomarkers, there's no doubt that we didn't solve the problem today, but we had a solid conversation that I think brought us together in a similar manner. And likewise, the same with the COA measures, what we should be doing, how we should be, you know, working on those and pushing those forward for the community, for drug developers. Because again, like we said, it's all about the patient and how do we get these therapies out to the patients as quickly as possible. I don't think we spent enough time on AR, but we'll save that for the consortium as well, because clearly we need projects within the autosomal recessive form of this disease. So again, thank you, everyone. We're going to be moving to our break, and we're just about right on time, so that's perfect. We will see everybody at noon at US Eastern Time. Thanks a lot.
Housekeeping items. We just want you to know all participant lines, you are muted. So if you are not a panelist or a speaker, you will not be able to speak during the event, but please use the chat feature if you have comments to send back and forth. We do ask, though, that if you have questions for the panelists during our panel discussion, that you use the QA function. And you'll see that next to the chat feature, those 3 ellipses, if you click on that, it will bring you to that QA feature and you can send questions. Also want everyone to know that the meeting is being recorded. So this will be available on the CPATH website after the event. Just give us a little time and we'll have that posted in a couple of days. So be aware of that. And then last but not least, if you need any assistance at all, technical assistance, or have questions that you need to ask, please send those to Kitty Boji or myself, Wendy Vanasco, and we will make sure to address those as quickly as possible. And with that, Frank, I'm going to turn this over to you.
Thank you very much, Wendy. I hope everyone can hear me. And I'd like to ask Romaldis Michaelitis and Alisa Thompson, my co-moderators, to go ahead and share their video. And I am going to just introduce the topic until we get to our patient perspective. But thank you both for agreeing to co-moderate the session with me. Definitely appreciate it and all the work you're doing. Just for the next slide, we're going to go over what it is that we'll be talking about this afternoon. We have just this brief introduction before we go into a patient's perspective by Peter Cody Fiducia. And then we will go back into sort of a working session where we're going to combine both the panel discussion and sort of a brief introduction to what we have done and what was recorded in the earlier recording by myself and Ron as to what we were doing for preparing for a standardization of a protocol or an approach to studying PKD in clinical trials. The video presentations that are appropriate to this include just the summary presentation that Ron and I put together based on a number of working group discussions and the patient perspective. So I do hope that you've had an opportunity to look at them all. And I have to thank my fellow speakers for their participation in that, Ron in particular, and Greg, who will be sharing some of his perspectives on the disease and on what it is that we are doing as a group. So thank you all for that. Moving forward to a live patient perspective, however, Peter Cody Fiducia, and I hope I'm saying that correctly, will next introduce his perspective on what we're doing as a consortium. So Peter, let you take it away.
Thank you so much, Frank. And thanks everybody for having me. I assume everyone can hear me. If not, send me a quick chat. Okay, I see some nods in the chat in the video box. That's good. Having taught now 2 semesters entirely online at Zoom University, as we colloquially call it, I learned to look out for the nods in the chat to make sure that I'm not boring anybody. Okay, so thank you again for having me. It's really a pleasure to be here to talk about my patient perspective in terms of PKD. I want to first start by thanking all of the medical practitioners and the foundation staff, trial participants, pharmaceutical and research partners, and of course, everybody else for being here today and broadly for all the wonderful work that you do that is so important. I also want to thank Carrie and Greg. Greg, I viewed his pre-recorded video, and Carrie this morning for her very wonderful and touching story. I think that's incredibly important to hear from all kind of perspectives and sides, and hopefully I can add a little bit of context and information to all of your incredible work. So introduction to just me and who I am. I'm currently a PhD candidate at Cornell University. So greetings from today, at least it's sunny Ithaca. Anybody who knows Ithaca weather will know that it changes pretty quickly here. But my parents will probably describe me as a forever student. I've been in school forever, right? So definitely an academic. A lot of my research surrounds local regression modeling and education policy and data visualization. So from a quantitative perspective, I can follow along with most things, although I kind of lose some elements at the biological and medical end, but I'm very glad to be a part of the conversation. So I'll start with, as Carrie did, an introduction to my PKD journey. I also want to touch on some similar threads from Greg's video as well as Carrie's session from earlier today. So some initial similarities, particularly with Greg. My mother also has PKD, and it was a novel case in our family medical history. So to our knowledge, there was no prior incidence of PKD, and my mom had it, or my mom has it, I should say. And I found out when I was young as well. I can't exactly remember the circumstance, but there was some blood in my urine and we went in for an ultrasound, and that's when obviously we found out that there were some abnormalities. And I don't know that I was exactly diagnosed with it at that time, but that's when we found out that there were some significant abnormalities with my kidneys. From that point, when I was around 10 years old until, I would say probably the early 2000s, there was really nothing that happened in terms of an intervention. Mid 2000s, we were made aware of a study, a clinical trial being run out of a few different sites around the country, and that was the HALT study. So my mother and I were very fortunate to be selected to be part of the HALT study. I tried to find the paper from the HALT study to make sure that I could corroborate my results. I'm not sure if this is correct, but I know that we were one of the few, if not maybe the only, mother and son participants in the study. I could be wrong. Maybe I'm just making myself seem a little more important in my head, but that's how I remember it. But I know it was a very unique experience. And we were on that study until its completion, which, as you all know, included imaging and blood work, the telmisartan/placebo, and I also both of us were also put on lisinopril to manage blood pressure. So that was I'll talk a little bit more about the trial experience later, but overall it was an incredible experience. It was nothing but positive feedback and results and experiences throughout the entire timeline. And we were very fortunate to be able to work with an incredible team of caregivers and researchers out of that study that made everything seamless from beginning to end, which again, as I'll talk to later, is a very important consideration in my mind when it comes from patient perspective on trials. So much like Greg described and much like Carrie described, there was a moment for me that was the get real moment that kind of started to put everything in perspective. And that was for me in late 2019. I had been in my PhD program for a few years, and I started to do local checkups with a new nephrologist in Ithaca here locally. And he noted that there was a pretty significant decline in my GFR from the last time that I had it checked. And whether it was the tone in his voice or maybe just kind of an intuition on my part, I knew that this was something serious, that something was going to change. And I knew if I had the opportunity to speak with some of the individuals who had been so supportive and informative in the HALT study, that I would like to take advantage of that. So I spoke with a bunch of different individuals, and a plan was put into motion to see if I would be eligible to be put on tolvaptan. Again, the get real moment was made even more manifest by seeing my most recent MRIs, my imaging, and as anybody who has the disease, and certainly all of you who have so much experience with the disease know, it's a pretty jarring experience to visually witness someone with large kidneys with a lot of cysts, and even more so when that person is you. And so I found out that I was in stage 3B, and which was actually at a worse stage of decline than my mother, which was a little surprising to me, probably not surprising to those who know the disease intimately, but to me it was, because my mom is obviously older than me, has had the disease for a longer time. So it was, you know, a bit of a shock. And I think more than anything, as Carrie described, the most difficult emotional part during that period of time was seeing my parents' reaction to the current stage that I was in. And then I think more than anything, seeing my mom's reaction and trying to understand her potential feelings of responsibility or guilt or anything like that for passing the disease along to me. And though I'm not a parent yet, probably a little ways off for me, it certainly was, again, reinforcing this notion that there's a burden, there's this responsibility, there's this emotional context to the disease that's very powerful, and that can cause you to think about different things with regard to your experience. So thankfully, I was selected to be eligible for tolvaptan. I've been on tolvaptan since 2020, January of 2020, on the high dose. All my functions have remained stable, which I'm very glad. And I'm happy to have to be drinking about 2 gallons a day. I usually average about 2 gallons. Forgive my lack of conversion to liters, but I just go by the things that I can carry in my hand. So before I get into the aspects of the trial and the patient perspective, I do want to again touch on some shared concepts. There's a very real recognition of being placed in this position of emotional limbo, right? When we talk about the emotional and mental effects of PKD, it's a very interesting place. And again, I'm probably preaching to the choir here, but from a patient's perspective, it's interesting because we all know it's serious enough to warrant invasive procedures, right? In the best case, you get a kidney transplant, and in a not so best case, or at least in a maintenance case, you get dialysis, and both of which are very serious. They're invasive procedures, and they change the way that you live your life. But simultaneously, the disease is not necessarily, in most cases, at least in my own experience, not aggressive enough, or not, I guess I'll say aggressive enough to warrant the stereotypical, often dramatized on television series, you know, you have X weeks to live, right? And so it's a very interesting place for, at least in my mind, for patients to be, because in some regard, if it was serious enough where it's your time to expiration is 5 months or a week or something like that, it almost gives a sense of finality to the disease, which in some might argue gives you a sense of control. But at the same time, without that endpoint, knowing that just in the future, as Carrie said, at some point in the future, my life is going to inextricably change, is a very difficult concept to wrangle emotionally. And as Carrie mentioned, these 2 times a day that we take these beautiful blue pills that we're so thankful for are in essence a reminder that at some point in the future, which we can't exactly tell you specifically when, your life is no longer going to be normal. And finally, it gives you this sense that it puts things into perspective. I realize that having these thoughts, I shouldn't be too, I should be thankful, right? I'm on a medicine that's managing my disease well. I have a talented group of caregivers and practitioners that are helping me through. I have a supportive family. I have wonderful scores of researchers and foundation individuals who are caring about this disease. And so it's a very interesting place to be mentally, but very thankful for all of the different modes of support. And then again, this notion of strength in perceived weakness, as Greg mentioned in his recorded session, I would just characterize that as strength. This community has a lot of strength and resilience. And I'll share a very brief anecdote for those who might be familiar with the social communication site Reddit. There are what's known as subreddits or communities that focus on particular causes or interests. And there's a PKD subreddit. And I've kind of made it my goal, I guess you could say, as part of the time I have away from my PhD studies, to post and to reply and to like and to support as much as I can on the PKD subreddit. And the amount of times that I've commented, don't worry, it's okay to go to the bathroom every hour if you're on tolvaptan. You know, don't think of it as a negative. Think of it as a side effect of a wonderful therapy that's allowing us to have a more stable life when it comes to PKD. So it's a wonderful community, and I think we have a lot of strength within it. And then finally, I'd like to talk about this notion of trials and the patient perspective. I wanted to frame it in the sense of some questions that may be helpful to the group here today. We talked about what matters to patients. I think delivery of information is crucial. How is this potential therapy going to help me or others? When would this new therapy be available? And then similarly, are there other maintenance therapies that can be kind of implemented? So for instance, when my mother and I were on HALT, we were also put on lisinopril, which was, to me, I frame it as a maintenance therapy because I was not on a blood pressure medicine prior to that. And I had the additional benefit of being put on that drug, which was obviously a help, as we all know, to maintain blood pressure. And what do we want a therapy to do from a patient's perspective? Well, the ideal scenario is to rid us of the cysts and the problem, right? We all know that. But in lieu of magic beans, maybe the best way I can describe it is a stabilization. We want to stabilize the decline of the kidney function or to slow the cyst growth. We talk about the reassurance of safety. I think that's highly subjective. I think it's also stage dependent. You know, I would personally be willing to take on more risk in terms of safety, because if it's a therapy that might help, I would be willing to take on that risk. I don't have a family, I don't have kids. I should say a wife or you know, a significant other. But I think it's very subjective. And then this notion of how patients deal or feel with being in a control group. I would say, again, it's very subjective. Something like a study that Carrie described where you're on tolvaptan, you probably know one way or the other whether you're on the control group or not. Something like my mother and I were on with HALT, maybe less so. You may not know immediately. And then finally, very briefly, the patient-reported outcomes. I think it's excellent to hear what worked and what didn't work from patients. So I think we should increase the kind of frequency with which we report out patient outcomes where privacy is eligible to do so. So in conclusion, I do want to recognize that my opinions are informed by a unique set of circumstances. I have a higher than average educational background. I'm not from an underrepresented racial group. My family background is about average socioeconomic status. So recognize those limitations with regards to my patient experience. And the only thing that I can say that I'd like to see happen or bring to the table here from my experience in academia, translating between technical and non-technical audiences, is to be straightforward, to be upfront, to give as much information as possible, and be ready to discuss, anything and everything that could be a potential positive for patients. So I want to thank you again for the opportunity to speak and run through a lot pretty quickly. I'm really looking forward to the panel discussion later. And thank you to all of you for your dedicated and steadfast work in making PKD, hopefully in the future, a thing of the past. But thank you so much for the opportunity to speak today.
So Peter, thank you for your perspectives. I think it's very, very important for us to hear, from the different patients that we've heard from today and yesterday. Everyone has a different take. As you said, things can be subjective, things can be personal, and I think you've helped share some of those aspects. You've also set forth, the objective, the goal for us, and in terms of what types of things we should be looking for. But I'm hoping that during the discussion that we'll have in the next hour or couple of hours, that we'll also talk about the path of getting there. And then once we get there, as you said, how do we share information back to patients? And you've also highlighted in your experience of looking at your MRI that it's important to ask the patients if they want to know something, as opposed to just sharing it with them. So while we can try and be transparent, I've heard from physicians, around the world and in different countries that have argued either for sharing such information or for not sharing such information. And I don't want to be archaic like we used to be in the old days of not telling a patient, you have cancer, because it would, worry them. But I think taking the time to understand where a patient is in their disease and their acceptance of their disease, and then modulating the conversation appropriately would be helpful. So I'm looking forward to your active participation as a panelist, and as we go through this, and as well as some of the others panelists that are representing the patients, we want to cycle through your perspectives as well as the regulators and the trialists and others. So thanks again. So with that, I want to go ahead and formally begin the next part of our session, and it's going to be mixed, as I said. We're not going to reiterate everything that was discussed in the recorded session by myself and Ron Ferrone, which was, but I will tell you what it was about and how we got there, and sort of the top line summary. Again, I want to thank my co-moderators, Aliza and Ramaldis, and then all of the participants in the panel today. And perhaps since there aren't that many listed, maybe we could take the time, if you want to go ahead and share your videos, and we'll go through the list just so that people can be introduced, those of you we haven't heard from before, and put a name and a face potentially to a voice. So you know who I am. I think we've heard already from Aldous. Carl, do you want to introduce yourself?
Sure. Hi. This is Carl Kramer. I live in San Diego, California, and I work with Regulus Therapeutics. And we are developing one of the newer drugs for ADPKD. And I love hanging out with smart people, and I just can't wait to participate in this lively discussion. Thank you.
Thank you, Carl. We certainly know you, Peter, so we don't need to go over that again. But is Kevin on the line?
Yeah, hi Frank, how you doing? Hi, my name is Kevin Fowler, and first of all, I'll say thanks for the invitation today. Just my background, I actually professionally, I work with companies developing treatments for earlier intervention of kidney disease or transplant. I live in Chicago in the process of moving to St. Louis, and then I've been living with a kidney transplant since 2004. And I also want to thank everyone for having this meeting today too. So looking forward to it.
Thank you, Kevin. Ken.
Yes, hello everybody. Ken Grushallo from Health Canada. I was the lead reviewer for tolvaptan submission for the ADPKD tolvaptan submission a few years back that led to its approval. I have an interest in the disease. My current work has me reviewing a wide array of submissions across various therapeutic areas. So I have some context for where the ADPKD general clinical development is. I think it's a very exciting time. The future is staring at us in the near to midterm, and it's just a matter of working through things carefully, diligently, walking through the looking glass and entering the era of what I think initially will be stacked therapies. In other words, 5 to 10 years out, I think there's a good possibility we'll have multiple therapies that will be used one on top of each other, add-on therapies, obviously with patient selection. And that could be very exciting. And in other therapeutic areas like heart failure, for example, has resulted in really massive improvement in clinical outcomes over the last 10, 15, 20 years. So thank you for, I look forward to this panel.
Thank you, Ken. And I'd like to remind the other panelists to go ahead and activate your videos so that we can see your smiling faces as well. Tess Harris.
Yes, hello everyone. Good afternoon, good evening. I'm from London. I'm Tess Harris, the CEO of the PKD charity. I've been working with the charity for 15 years now, and I'm an ADPKD patient, got a transplant last October. I'm active in the UK and in Europe. A few people on the PKDOC know me, having attended several events over the years. I was also on the EMA working group looking at the qualification of the biomarker in 2015. Sadly, I missed out on Tempo and Reprise due to age and EGFR, but I would have taken it at the drop of a hat. But anyway, I'm really looking forward to this session, and thank you very much for inviting me to be a participant.
Thank you, Tess. And actually, we'll address that, I hope, during our panel discussions about the inclusion and exclusion criteria and why we do it and should we be doing it that way. Neil, you're next.
Thank you, Frank. I'm Neil Schusterman. I'm an adult nephrologist who used to practice at the University of Pennsylvania here in Philadelphia. But for about the last 30 years, I've been a drug developer at various companies, and currently at Palladio Biosciences, where we are developing lixivaptan as a treatment for ADPKD. And I'm glad to be part of this panel. Thank you.
Thanks. I'd like to welcome my colleague, Lerone Walsh.
Hi, thanks, Frank, and appreciate the invitation to join this exciting panel. I'm Lerone Walsh. I'm an adult nephrologist as well. I lead clinical development efforts at Goldfinch Bio, and we're interested in really bringing precision-based therapies to patients, including ADPKD. So thanks again for inviting me.
And finally, Alan Yu.
Thanks, Frank. Thank you for inviting me to this panel. So I'm Alan Yu. I'm a nephrologist and clinical researcher at the Jared Grantham Kidney Institute and the University of Kansas School of Medicine.
Thank you, Alan. I'm going to go over a few additional slides just to set the stage and to familiarize those of you who may not have had a chance to look at some of the pre-recordings. And then we will get right into it. And as I said before, what I would like to do during the discussion portion of today's topic is to cycle through first with my co-moderators, Elisa and Romualdus, some of their perspectives on the questions, maybe include some of the panelists after they've spoken who have a regulatory or industry perspective, but definitely want to include the patient perspective. And so I'm looking forward to hearing from Peter, Kevin, and Tess in that regard. So in the next slide, really it sets the standard or the objectives for what we are trying to do today. We really had hoped that we would be able to align our expectations from the perspective of ADPKD trials, what might be best practices. Really, it's a discussion and it's a formative exercise at present. In the long term, however, we would very much like to see, just as has been done in oncology and for many cardiology indications, that a standardized approach to conducting clinical trials in PKD could be adopted, and not only adopted, shared across industry, across academia, so that it would be possible to make the patient contributions, the 4-hour round trips that we heard about earlier today, a little bit more cost effective for patients, so that we would make trials more efficient, make delivery of drugs to patients faster, and with a better understanding of both their efficacy and safety. So what we have done is consider the methodology, consider the endpoints that are the key to our clinical trials, and try to sort of center the discussion around them, around how to best use them, about some of the concerns or uncertainties with regard to their use, and how to capture all the data that's valuable, including patient-reported outcomes, which we talked a lot about earlier this morning. How we did this was to involve a number of working groups, and I'll go over those shortly, to discuss these gaps, discuss these particular endpoints, and come up with sort of their thoughts as to what we should do, what we should propose, and then summarize that in the recorded session that is available to you all, but that was performed by Ron Perone and myself, to sort of summarize some of our perspectives on a clinical trial. In the next slide, sorry, I've had too many different controls here. The working groups, as I said, consisted of a number of individuals, including session leaders from academia. We did not include regulators at this point. We wanted to involve the regulators more so at this point to get their feedback, their perspectives, and their advice in terms of what was discussed and what else needs to be discussed or done to potentially use these tools toward a trial that can be standardized. And then in my last slide, before we get to the discussion, we presented in that summary presentation a study trial design proposal. And really, this is just a list of probably the most important considerations that were made during the workshops around what the objectives, endpoints, populations, and trial durations should be, but also some important concepts for us to discuss. And we'll be discussing some of these here today. As Tess mentioned, do we focus on broad enrollment or focus on enrichment of trials so that we can get the biggest bang for the buck and the fastest, most efficient trial done? And the answer is quick as possible, and hopefully an approval as quickly as possible if the data support it. These are concepts that we see exercised all the time as different industry and academic groups put their trials forward, but their objectives may be different in some cases, and so their designs may be different. It's possible that it would be impossible to create a standardized protocol that would cover everybody and all aspects of PKD. But we think that there is at least a foundational core that should be shared across these protocols. So looking at the box, the blue box on the right, we believe that the objectives really should be to evaluate the effect of treatment on progression and in consideration of long-term safety. So as Peter had said, we really want to, if possible, put the disease into stasis, if not reverse it. and that should be an objective of most therapies for PKD. There was a general concurrence that for now, GFR and TKV may be primary, and primary in different contexts, meaning that GFR is much more closely related to the main outcome of interest and maybe considered an intermediate endpoint depending on how you look at it, while TKV may still be more of a biomarker surrogate. But we also thought that it was important that PROs or the patient experience during the trial is important to put all of that into context. We talked a bit about estimand. Endpoints are one thing, but estimand with all the considerations of how you collect that endpoint is really, really critical. And we felt that for most studies that are aiming towards registration for an interventional product, you're going to be focused on patients that are rapidly progressing because they are the path of least resistance, the means by which one could most quickly assess the effects of the drug. And so when you design a trial, you will probably create an analysis plan that includes or segregates only those patients that have rapid growth of their kidneys or rapid GFR decline or both. And you may look even within one trial at two different populations or within one program at two different populations. We'll talk a bit about pediatric research and the challenges that GFR has in that population since they're basically all normal, and the possibility or potential for using kidney volume or other surrogates in those patients as a confirmation of a potential beneficial effect going in the right direction. We talked a lot about what was previewed in some of the videos earlier this morning about the Mayo class classification for TKV efficacy, about how we look at patients with different degrees of CKD for GFR efficacy, what to look out for when we're measuring these things. We talked a little bit about what it would take for power and what types of populations might be ideally used for evaluation. But as Tess said, if we draw a line at age 50 and say you're not going to treat patients at age 50 with the drug, you're not going to understand what happens when a patient who's been on the drug reaches age 50 and continues. And, what safety in that population of patients, we really wouldn't know. So this is sort of the schema or the end result of many of the things that we've discussed. And I encourage everyone to review the summary presentation. And at some point, we may also have the hour-and-a-half sessions that made up each of the components where we looked at CKD, where we looked at TKV, GFR, TKV, and other endpoints like pain separately. With that, I'd like to, again, welcome Eliza and Romualdus with the first question for discussion. And that is, how can we encourage both industry and academia to and toward a more collaborative effort, the development of master protocols, of umbrella type protocols to facilitate research in ADPKD? And let me turn first to Eliza, if you wouldn't mind sharing some of your thoughts or views on that.
Yeah, so I think you're starting off with maybe the hardest question for me, and I'll be very interested to hear from Romualdus and Ken and certainly our industry colleagues and others for their views on this matter. There has been tremendous interest in master protocols, and clearly, perhaps with those great experiences and on the oncology space and in COVID-19, I would argue both relatively well-funded spaces. And I would say that obviously these types of protocols and, specifically, as I understand, you're proposing more of an umbrella type of trial. They have a lot of advantages, but they also are associated with some challenges, certainly require a lot more upfront planning and coordination. And so I don't know really from a regulatory perspective, what to say right now, other than I think any therapeutic area should explore this as an opportunity in whether this is something that's right for them, and consider building the infrastructure, but also know that a lot of I think the needs in this space, in terms of facilitating the design and not so much the design, but the conduct of these trials. I think there are other ways to look at that too, in terms of, decentralized trials, also other means to consider how to make this less burdensome to patients and really open this up to a much broader patient community than perhaps we've pulled in the past. So again, look very much forward to hearing other people's comments on this issue. And of course, would be receptive to this if this is where the community wants to move.
Thank you, Lisa. Let's move to Romaldis and then maybe end so we can get all of the regulators' perspectives.
First of all, I agree with the bullet points you summarized nicely. What is a challenge for anyone developing a product? That's why industry should agree on all bullet points. First of all, I would be surprised if the academic people would not be interested in joining this novel sort of trial design, logistical challenge to learn and to explore it in different areas. For the industry, I think that what is really important, firstly, and I agree with all those points, but I could also emphasize another bullet point, that what is always important to payers, at least. At the end of the day, they are deciding to pay or not to pay. So country from country differs. At least in my country, we don't usually see that, especially in the common HTA SOP meetings, that they don't care about the non-feasibility. Okay, you have a patient, yes, you cannot tolerate tolvaptan, so we cannot, we don't care. Give me the number. And this is a real challenge for anyone. And is it big company? Is it small company? As I see from the list, what we invited here in our discussion, there are not all big companies jumping like it would be in COVID vaccine area. So it will be challenged. So this is opportunity. I don't know whether it is for confirmatory purpose opportunity to be explored or for more exploratory to understand. This is something else to discuss because to have direct comparison for the standard of care, let's say for the tolvaptan, I don't think this would be okay. This would be one of the options. But as far as you go to earlier disease and you see another niche and you see the need for placebo, then of course you will not comparing. You will not be able as much as I'm aware against the so-called standard of care. We will come to this in the next question. But at least this is a real challenge to anyone developing these things. And the last thing, what I think we came several times during those 2 days, is the choice of biomarkers for these umbrella trials, which to choose. Because we heard inflammatory, we heard some other things proposed, and nobody is sure which will be the best to enrich, to find the one or another group in this umbrella trial. So I think that when you have the type of developments, I think you have big level of discussion and agreement. Of course, they should be always, as it is clearly mentioned, even also in FDA guideline, it should be IND set type of discussion. So similarly with us in SALT. So then you have quite, early set of the promising biomarkers to split your your ADPKD into several groups to test your product on several subgroups for enrichment, for whatever. So I think that there are a lot of important things which are difficult to meet on their own in these areas when you have only, let's say, best option to have standard single arm trial like oncology. But we should and companies should realize that this is an option. So I think that this is from my side.
Thank you, Romualdus. Ken, why don't we get your perspective, then I'd like to open it up a little bit more to other perspectives.
Yes, thanks, Frank. So I think I certainly applaud the notion of reducing clinical drug development for ADPKD that includes potentially different patients at different stages in their PKD trajectory. And data that's generated is good, which goes without saying. However, in order for this to pass regulatory muster, it's quite a complex process, and one needs to be aware of that. Go in with your eyes wide open. One of the first comments in terms of specifics is you've used different terminology here: umbrella trial, master protocols. They're not all interchangeable, but that perhaps reflects some of the challenge of even framing this approach. In addition, obviously, notionally, this is quite attractive, perhaps, for drug development. But in practice, I wonder if industry, when they have a drug they want to develop, obviously this is a critical question for them, but my guess is they'll probably gravitate based on the drug and what they think they can show to one of three different stages. One, in talking about ADPKD, it might be pediatric with very advanced rapid progressors that are identified. You might want to identify early on post-pediatrics, so that's an early stage intervention. And then there's sort of a tolvaptan stage, mid-range. So it may, obviously this is an attractive issue to consider in general and obviously critical for the sponsors, but it may kind of sort itself out to some extent. So that's, but the notion of moving ahead on this front, you really, to pass regulatory muster, you can't just generate a lot of very interesting and worthwhile data across the board and then expect an approval. Obviously, you need to pre-specify your processes, your endpoints, and all the rest of it. So this has to be pre-specified, even if it's a complex master protocol with different, and that complicates things markedly, let alone before we even get to the appropriateness of what endpoints you're going to use as a primary endpoint. So it's quite complex. I'm not throwing cold water on the notion because I love the underlying notion, but I think you have to go in with your eyes wide open. The last thing I'll say is, we need to be thinking of what we can do now or in the near future. And I think one way of proceeding on this in relation to this issue is to think of providing guidance, like a framework, a document, developing a document that would be a framework for developing clinical trials in ADPKD at different stages and what issues and aspects should be considered. I think if that could be developed with regulatory input, not necessarily obviously having regulators signing off literally on it, but just notionally on the approach, that might help to better define reasonable approaches for industry on a go-forward basis.
Thank you, Ken. I think your and Romualdus's comments about different stages or maybe different phases of research in terms of where this might best fit are very helpful, and potentially giving the group and particularly the Outcomes Consortium some advice in terms of what might be the first easy steps and what might be steps a little bit beyond our capabilities. So thank you for that. I noticed may have a question, and I'm going to open it up, I guess, to academic perspectives, but I don't want to forget, before we leave this topic, I would like to hear from the patients and the patient representatives. What?
Hey, Frank, if I could comment, I just think that part of this is like if you look at these master protocols and umbrella, it's really patient-centered. I just think the biggest challenge is just the education. There's such a gap of education. And I know the patient organizations are loaded with a lot to do, but this is just an education gap from my point of view.
Oh, thank you. I appreciate that. And I apologize. Someone said my audio is cutting out a bit. I hope it will work, but I don't think I can hear from Fuad, and it may be because he's not on the panelist session. So if you could type your question, it would help. I do see there is a question from Norman, Dr. Stockbridge. He asks if we could explain, I guess it's in the previous slide, so let me go to that. What is meant by powering for each endpoint. One of the things, and I'll explain this briefly, but hopefully there's a little bit more detail in the summary, and there's definitely detail in the conversations that we had. What we were talking about here was if we had a very expansive population being studied in the trial, meaning that we weren't restricting or trying to enrich a registrational program or a registrational trial specifically, that we might have one population or subpopulation within a trial where TKV was the primary endpoint, where we knew that we had the power for patients that were Mayo Class 1, E, D, and possibly C to test the hypothesis that we could slow the progression of those rapidly progressing patients. They may not be patients whose GFR is progressing. In particular, in pediatrics, you wouldn't necessarily expect to see any effect or any potential impact over a relatively short period of time on their GFR. So you may look at TKV early on in younger or earlier stage patients and may focus on GFR later, but you would try to collect both endpoints in both so that you understand the context and the relationship overall. But for the S demand, you would make it very specifically the enriched subpopulation where you think you could have effects. So I hope that addresses that question. Let's see. Were there any other questions? I don't know if Fuad had a chance to comment. And if not, let me open it up to any of the other panelists about this topic, patient or academic or otherwise. Let me go back to the question, excuse me.
So Frank, if I may
It would be great to hear from industry as well.
Yes.
Frank, it's Tess.
Yes, Tess, go ahead.
I was thinking about your other slide again, actually, which I realised that you're looking a bit forward, but I just wanted to welcome the broad approach, and as I think it was Ken outlined, looking at the spectrum from pediatrics with a particular biomarker versus the early stage where TKV might be more appropriate, and then the EGFR in the later stage. And I think that's certainly would welcome that, and it would hopefully allow a broader inclusion criteria. One thing I did notice you haven't really mentioned is genetics. It seems to have sort of disappeared out of slides, and I know having seen some of Peter Harris's work, it's not the be all and the end all that probably was thought to be, i.e. if you've got PKD1, you're definitely a fast progressor. But we know there are degrees, as indeed in my family, with extreme variability in PKD1. But increasingly in Europe, patients have, I guess in Western Europe, I'd say, patients are increasingly getting access to genetics, and that will be added to their health records and could also provide another way of enriching a population. So I wondered if there were some reflections on that. I think, and you're forward-looking in terms of the umbrella. Elisa made the point that it would require massive planning, and I would worry that it might slow down some of the trials that are in progress, for example. And for some people, we then go past the point of no return and are deprived the option to take part in any trial, whilst months, maybe years of planning have to take place to enable an umbrella trial to go ahead. Let alone, the complications of getting a master protocol agreed everywhere across different jurisdictions, etc.
So all very good points, Tess. I appreciate it. The panelists, or the working groups, I should say, did talk quite a bit about genetics in the context of what would be the right way of enriching a program for TKV endpoint or GFR endpoint, et cetera. And while there's a potential for it to be to add to some degree in terms of the selection, it wasn't viewed as being as critical potentially as having a good height TKV assessment of the individual patient at that time. That being said, and I'll try to summarize, I believe that the consensus was that genetics should be collected and if possible, reported out as a factor in, whether a particular population or subpopulation of patients may respond better or less well to an intervention, but it wasn't viewed as something that would be critical, but something that should be collected. In terms of moving towards the umbrella trial approach, again, we don't know when is the right time or where is the right time to do this, but we're hoping that through the consortium, we might be able to find a good pathway, and we recognize that it will take many years potentially to put these things in place or add value through some standardization. But we definitely don't want to slow anything down in terms of what's already ongoing. So any other comments?
This is Neil. I just had a couple of thoughts, if I could.
Sure.
I haven't followed this area in oncology, I have to say, but I have been fascinated during the COVID pandemic of these types of trials that use multiple therapies and randomize individuals to those therapies. And it just occurs to me, and I'm sure it's occurred to this group and you, Frank, that this is a really highly efficient way of doing business and getting high quality, uniform information. All of us know multiple companies spend lots of money, lots of time doing their own trials, sort of like in their own silo, trying to get the limited number of patients that are out there. And this whole idea has a lot of merit and a lot of positive value. But I think, as Elisa pointed out, also a number of potential hurdles. A couple that just came to my mind in looking at this design. How do you blind across arms if you have to do a blinded trial? How do you align if, say, different therapies are at different stages of development? The nice thing about COVID is that most of the things that were tried in these umbrella type trials were established therapies, so you could get your hands on them easily at the same time. And hypothetically, can you actually do pairwise comparisons of, drug one, say, against control, without having to take a penalty for multiple comparisons? So I think there's, obviously a lot here to think about, but also enough upside to give it more emphasis. And I'm glad that the consortium is taking this on.
Thank you, Neil. I know there have been several additional written comments, one from Jerry Kolka about using umbrella trials in phase 2. As I think some of you have mentioned, a lot of companies are studying their drug in a basket way, where you take one drug and study it across a number of different indications. maybe, and that's very often done in phase 2 to sort of see where the effects might be most evident. Maybe this is something that could be done for phase 2 for early, and help extend companies that are thinking of a drug for a primary indication to sort of easily test it in PKD if this was available to them. And I could think of, for example, SGLT2 inhibitors. I don't think anyone's done a formal study with those agents, but tens of thousands of patients have been studied for a variety of indications. It would be lovely if this could facilitate something like that. We have a comment from Peter Harris, who's throwing his support behind genetics, just as tested, so appreciate that. And then another comment from Michael Schenkel about excluding patients from the trial and what that does at the end of the day in terms of payers including or excluding them from the availability of the drug. I think those are all important contexts. If there are no other questions or comments on this topic, I think we can move to the next question.
Frank, could we
hear
from Alan Yu? I think Alan wanted to say something earlier, and I'd be interested in his perspective.
Sure, absolutely. I didn't. Please go ahead, Alan.
Thank you. Thanks, Ken. So from an academic perspective, I think we'd be strongly supportive of this type of approach. And I should mention that this isn't just confined to the oncology and COVID space. There are, I believe, at least a couple of consortia in Australia that are doing this, academic consortia that are doing this successfully in the CKD space. So it's doable, but my understanding is that the amount of infrastructure needed is quite substantial just to get started. I also just wanted to comment that I think it would be terrific to have a type of structure that would allow direct treatment comparisons. And speaking as a clinician, I think it's always a challenge that we have a lot of drugs that are approved for treatment of diseases individually, but no one ever does a subsequent comparative efficacy trial, and it's hard to do after a drug's approved. So that's certainly something I would be strongly in favor of.
Thank you, Frank, can I just go ahead?
Hi, it's Lauren. Yeah, just with respect to direct treatment comparisons, while there may be a potential in this sort of trial, there's probably some statistical hurdles to sort of doing that, I think, as Neil had mentioned. But I think we can agree that there's likely not a single agent that's going to cure PKD or halt TKV or prevent further progression. So even if one is numerically better, T1 compared to T2, let's say, there still might be value in adding T2 to a patient's regimen. So I don't think that industry should necessarily be averse to this sort of design with the concerns that what will happen if my drug looks worse than somebody else's. Having said that, I think there are hurdles that Neil certainly points out, and Ken in particular, with respect to standardizing endpoints, the collection, data sharing, and I hope that in the future that both patients, academics, and industry and regulators can get to a point of providing a framework to participate in something like this.
Thank you, Laurent.
Right, and
go ahead, Elisa.
Just one, Frank, two quick things. One is one is a clarification request, and then the other is I was wondering if you could unmute Norman for a second, because he may have a comment or two. But just a clarification. So, if you look at how in recent guidance documents, how we define an umbrella trial, it's that all therapies are entering at once, which is different from a platform trial where, one therapy enters and graduates and another therapy enters and graduates. Are you in fact saying that all therapies are going to start at once, which assumes all therapies, essentially it requires everyone to be at that stage of development? I just wanted clarification on that. And then again, Norman has a comment if you can be unmuted.
Yeah, so for clarification, no, I don't know how it would work. I don't think we the details are specific how it would work. But But your point is very valid, and it's something that I was thinking about as well. The figure that you see on the slide really doesn't say how it actually can be implemented properly, and I think the adaptive nature that this could also support would be very helpful. In other words, as drugs enter or are tested in this paradigm, maybe in phase 2, they may either succeed or not succeed. And so a play the winner or drop the loser type of approach may be very beneficial in terms of, further accelerating those drugs that are potentially showing a benefit so that they can get to a phase 3 study quicker, and sort of weeding out the ones that aren't going to make it so that companies don't expend time, patients don't expend, their efforts towards something that's not really going to help them. But always in the context of what's the best standard of care at that point in time. Is that address your comment or your question, Elisa?
Yeah, it does. And some of what you're saying makes me think that this is almost better for a space where you're testing sort of drugs that are generic and already approved, and you're thinking that it could work. And yeah, it may just, yeah. But anyway, I'll stop. And yeah, I think again, Norman may want to say something.
Yeah, I think someone did unmute Norm, so please.
Yeah, so I was interested in at least introducing the idea that if you've got a number of candidate treatments that have at least nominally different mechanisms of action, the way to learn something about whether or not they both work and possibly work together is to do factorial trials.
Mm-hmm. No, absolutely, and I think that would work in the context of what Elisa was saying, where maybe you have in the illustration you see there are 4 different treatments being tested against a control. You weed them out, but 2 of them seem to be doing something beneficial. Maybe the next phase of that is to combine them, to actually add both of those treatments to the same patient to see if you're getting or some randomization subset of those patients to appropriately test whether the addition of both drugs is further benefiting the patients or not. So absolutely, I think there are infinite possibilities that have been discussed. The problem is the heavy lift. And I don't think I'm not sure if we have any NIH representatives here today, but the NCI, I think, has probably been critical in helping oncology move towards this type of approach and this type of standard. I know that NIDDK, at least in the United States, and I'm not sure what the EU equivalent would be, but these governmentally funded and supported institutions might help create the foundation for this. I know that they do sponsor centers of excellence or areas for PKD research. This could be beneficial by establishing a network of investigators, a network that can operate in a like fashion with standard approaches and treat multiple patients with different therapies. Anyway, if there are no other questions, I can't really tell when people are raising their hands, but if there are no other questions, let's move on to the next topic.
Which I may just reply shortly about this EU. I think that our European Commission has quite a big initiative, Horizon and many others, which are pushing quite heavily for those type of collaborations. And I think that we have, if we could have one protocol umbrella trial with the issue of the feasibility. First of all, I'm always concerned about that, how many patients we can gather into T1 or T2 sufficiently representative against control, which is, we will go to that next question, not easy to interpret, which is very broad. So, and then you go to the numbers. So this type of multinational, Pan-American plus European studies, they are really very good candidates for the EC funded projects. Yes.
No, thank you, Romualdus. I'm going to give the last word to Susan Mendley if she's available. I think you've been added to the panelist list, who is from the NIH, as I mentioned, and responsible for the PKD centers. Susan, did you want to make the last comment on this? And maybe she doesn't. I'm not sure, but all right. Let's move on. I do want to be respectful of time, and I think we are running out. So I want to get to a couple of other very relevant and important questions. One that we discussed in the workshops was the comparator, and I think it's been touched on a number of times by different people. What is it that we should be testing, and does it matter? There was, well, I'm not going to say what the workshops concluded, but I'd like to open it up to, again, our regulators first to ask what their perspectives are in terms of, drugs that are approved in their regions and whether those drugs should be used as a baseline for all patients in these studies.
So obviously, and I wasn't sure if you were raising this issue, but one issue is what's considered standard background therapy and what should all patients be getting that you add your therapy to. Obviously, another question is, if you're going to do an active control, what's a reasonable active control? And this may be something, again, then you need to beat the other, active control. I'm going to focus more on the standard of care issue. You all are the ones that are experts in this disease. We aren't. But what I can say is that we need the results of the trial to be applicable, and I think this is true for any regulator. I think Ken may say something similar, not meaning his whole response, and Romualdus as well, which is we have to believe the findings in this trial, which is going to be international, are applicable to a U.S. population receiving U.S. standard of care. And so I think, from our sense of the space at this point, in terms of what U.S. standard of care is, it's true that a lot of, we do have a therapy now that's approved for the treatment of ADPKD, but it's also reasonable, and there may be reason to believe that some patients may not want to take it. And so we haven't taken the position that all patients have to be on tolvaptan in the trial. We think it's helpful if a sponsor get data both in patients who are on it and who aren't, just because it probably is going to be more applicable to the findings in the U.S. But I think the overarching concept for us is really what is standard of care, and whether, we're making sure that, especially bearing in mind there's a placebo arm, that people aren't getting undertreated, or again, that the findings in the trial won't be applicable to our patient population.
Great. Ken, and then maybe Romualdus.
Yeah, sure. Thank you. Well, yes, depends on the context that you're studying the drug, but assuming that you're conducting a trial on a new drug in a patient population that's a tempo-like population, the expectation would be that the standard of care would include tolvaptan, and that the new drug would be looked at in addition to. So a placebo-controlled trial with tolvaptan in the background. This is quite often done. It's pretty standard in the cardiovascular area, but there are some caveats. Obviously, it would not apply if you're looking at, for example, very early stage ADPKD. If tolvaptan is not the standard there, we would not expect that to be background, of course. So one has to think about what population you're treating and take that into account. So that's really the main element. The other thing is, if you are expecting or allowing for tolvaptan to be background therapy, of course, we don't expect 100% of patients to be exposed to tolvaptan. It would be a reflection of what is a reasonable standard of care at the time of initiation of the trial. Sometimes we get a result in a trial that's a registration trial. By the time the trial is completed and we're analyzing the results, there's a new standard of therapy that's been approved that wasn't included, but we see that pretty regularly. So obviously that's something that's a review issue. We take that into account. We tend not to penalize the sponsor for not including a new approved agent in a therapy that occurred after the initiation of the trial. And the last thing I'd just say is, if this is the scenario that we're looking at, then the question is, what is the efficacy of the new drug? It would be on top of standard care, which means some people had it and some people didn't. Let's say hypothetically it was 50% of patients were on tolvaptan. If the study design was appropriate right from the get-go, then what we would probably do for efficacy is look at the overall results for starting with the primary endpoint for the overall population, and then we would look at subgroup analysis for those with and without, and that would be pretty important. And we would obviously hope that the results on a relative basis would be consistent or reasonably consistent across tolvaptan treated patients or not. And of course, we would be looking at safety issues and all the rest of it, so a wealth of data. But this kind of general approach is pretty standard for in other therapeutic areas where there are different therapies being developed on top of other therapies being used in a similar fashion. By that I mean, same indication and long-term therapy typically.
Ken Romualdez?
We discussed during previous session that standard to trigger called tolvaptan, it might be different in Europe and in the US due to different approaches. What does it mean, rapid progression? So this type of differences base the need for a priori agreement on the standards for the whatever study you are applying and planning to have authorized not only in one jurisdiction but in another. And what is this? Okay, this is purely clinical question. Is it accepted as a CKD disease with some features, or is it just some only CKD, only polycystic kidney disease, not without clear relevance for the delay of progression for any CKD? So this is a local concept which should be respected, and this is the only thing what I can say for this question.
So I appreciate all of your comments. I think the answer is it depends. But if it does depend and a study is designed to allow for their use, not necessarily mandate it, appropriately stratify or hope that randomization, unbiased randomization would account for those differences. So you could do those comparisons that Ken talked about in the important subgroups. Let me open it up more broadly to patients and others. I mean, I'm assuming that, and I think we heard this before, that patients worry about being assigned to placebo sometimes. So is knowing that you're receiving or you have the opportunity to receive the best standard of care, but it's your choice, is that something that would be important in any of these kinds of trials? And I'm hoping we can hear from Tess or Peter or others who have participated in trials as well.
Sure. I think I'll ask a clarifying question here first before I give a response, which is the question saying, are you asking what would be the appropriate comparison group if you are saying, okay, here's a study, we're doing a study with this new therapy, what's the appropriate comparison group, 1, 2, or 3 here? Or are you also saying that 1, 2, or 3 would potentially be, for instance, a maintenance medication like I was on lisinopril while we were doing a study?
Yeah, it's a little bit of both, Peter. The question is, if there is a standard of care, is it necessary that everyone use it, like the lisinopril that you might have received? And then what is the comparison? Is it, in your case for HALT, it would have been lisinopril alone versus lisinopril with an ARB or with a placebo?
Right.
So that's the test comparison. But the point I think, or the question I'm really raising is, does it matter to patients to know that there's a possibility that they're going to be on placebo if their alternative is the best standard of care versus an unknown, versus something that's being tested?
Right.
Hey, Frank, or go ahead, Peter, you're ready to say something. I'll speak after you're done, so go ahead.
Oh, okay. Sure. I think that it really does matter in what scenario you're placing the patient, right? Because I would, and this is only from my own experience, but I would argue that it may not be an accurate or a totally unbiased control group to say that the standard, and to Eliza's point, the American standard of care is something like tolvaptan. I mean, I know that I'm making my career decisions based on where I can get the current level of healthcare that I have at my current job. Because if I don't have the current level, I can't afford tolvaptan, right? So I think that is one consideration. If you're saying, well, the comparison group is any individual who's receiving the standard of care, then that's a very selective group who can afford a therapy like tolvaptan, number one. But if it's, do I want to know as a potential trial participant, I'll speak for myself and say, I would want to know all the information, but the likelihood is that I would say yes to pretty much anything in terms of a trial. Like I would be the requisite, scientific guinea pig to say, let's get it done. But as long as you tell me the information, I'm on board.
See, I see a little differently. I think first thing I would say is that I think you'd want to make sure that the assumptions being made that you've engaged the community early on and you've asked their opinion on that. And from my standpoint, I think it's a gray issue. And I think that gray issue could come out early on if you're having those discussions, depending upon that risk benefit. So I think it's not black and white. I think it's very gray.
Thank you, Kevin. I am going to note.
I was just going to add in, just from my many years of talking to patients, I've not seen any resistance to being on placebo. I think people are desperate to take part in studies and trials, as has been evidenced in the coronavirus vaccination studies, with people flinging themselves into trials all over the world. I think the standard of care question is very complex. Thinking from a European perspective, not every country in Europe has reimbursed tolvaptan, and it's in some cases people can't even get MRIs because that's not reimbursed either. So that may be preventing access, and some people I know getting it too late. So I think that would require a lot of in-depth trawling through each country's definition of standard of care, coming to some sort of consensus.
Yeah, so a little bit to Eliza's perspective, what's the real world application? Are you testing it in?
But if I could just add, because I don't think it's quite as complicated as might seem. From a regulatory, or from my point of view, if you have an approved treatment and if you're studying it in the same set of patients, a new drug in the same set of patients, the default position would be that one would expect the trial to be, and this is in the context of a chronic treatment, used the same way as the other treatment, in this case tolvaptan. The default position is that you would expect the registration trial to be done on top of existing approved therapies. You can simplify things by saying approved, and tolvaptan's approved most places in the world. Now, we know that obviously many patients, meaning a significant minority, if not occasionally a majority of patients, will not take quote unquote this standard of care. And there may be a lot of very good reasons for that, reimbursement being a big one, not believing it, and some people don't tolerate the side effects, etc. So obviously you take that into account. And so I don't think any regulator would expect 100% of patients or 95 or 90% of patients to be on tolvaptan. But I think, as a guidance for industry, I don't think this is a wild card. I think that would be the default position. But again, it depends what population you're studying exactly and so forth. But anyway, we have it.
And just to close, I think this maybe is an area where the different regulators may be taking different perspectives on this issue.
Okay, so it depends, and it depends on where as well. So I appreciate that.
Okay.
I'm trying to be respectful of John Michael because we are now eating into his summation time. He did say I could do that a little bit, but we actually have a number of questions we haven't even approached. So I'm going to take the moderator's perspective, and basically, I will flip up the remaining 3 or 4 questions that we had, and not go through a cycle, but really ask if there's anyone who has an important consideration or important point to make. Oh, wait, Wendy's telling me we have another hour. Oh, it's only 1:23. Never mind. It's been a long day, guys. We have tons of time. We can keep talking and talking and talking. I'm very happy for that. With that in mind, with that reminder, let me make sure that others not from the regulatory perspective or from the patient perspective have an opportunity to talk. Does anyone comment from the trialist or industry perspective?
Could I take up some of that time, Carl?
Sure, Carl.
So the other challenging issue here is, okay, we've got a study and it's a multinational study, and we've managed to define the control, the standard of care therapy a priori. And in the trial, let's assume that it's standard of care plus placebo versus standard of care plus the active drug. We're more than likely over the next 5 to 10 years to have a situation where another drug is approved. And most study designs would not allow for that drug to be added to the standard of care because it wasn't defined a priori, right? But what it's going to do is it's going to change the dropout rate in a study that's where the treatment's given over 2 years. So we either just live with that and build that into the sample size of the study, or does anybody else have any creative ideas to how to handle that?
So
just a quick.
I'm sorry, Carl. You predicted 3 questions forward in a sort of a tangential way. I think we should get there when we start talking about estimands and, that consideration of switching to a new therapy or some alternative therapy and how we mitigate that. But anyway, I'm sorry, Elisa, I jumped in there. Go ahead.
No, I'll wait.
So I think we'll get to that question, and I'm looking forward to hearing from anyone about how to avoid missing data due to issues just like what you just discussed. Any other comments or any other thoughts on this particular topic? And now that I have more time, boy, I'm much more relaxed. Thanks for correcting my time warp. Let's move on then to another question, which has to do with total kidney volume and its use either as a likely a surrogate for outcomes of interest, but focused a little bit more on what's the effect size that you need to see for such an outcome, and how would that translate to benefits in terms of maybe the more desirable outcome like renal function or preservation or time from ESKD. So the question really is, if we're using kidney volume to further accelerate and encourage research in PKD, what do we need to think of in terms of its use? And in particular, this slide has 2 illustrations. One on the right from a group at Sanofi who I believe proposed that a 50% reduction in TKV growth rate may be associated with a certain reduction in rate of decline of GFR. And then the information on the left, which was in the supplement to the first TEMPO program, which discussed the issues around acute effects on TKV and how you must account for those or interpret an endpoint that may seem to be very objective, but actually could be quite complex. So again, let's cycle through since we have the time and hear from the regulators first as to this topic. Maybe we'll switch it around and not put it all on Elisa. Let's start with Romualdus this time, if you have any comments.
So let's start from the point that we had the famous NKF, FDA, EMA workshop on the surrogacy of GFR and the applicability of GFR, and we had a lot of publications, and we had a perception that by the community that this works. Seems GFR is working. I cannot speak for the FDA, but when we had discussions of what do you think about this several ml gain in the 2 years or 1 year, which is predictive to the end-stage renal disease, we always go on case-by-case basis for the decision. Yes, it might work. Well, it's not working because the population was not representative enough or some other reasons. So here is similar thing. It is difficult to comment the robustness of these, first of all, correlations, although a number of studies put toward that direction. When we speak in general, it seems similar to me that it might be working, this TKV growth, 50%, shows certain benefit. And but regulators, they are, love numbers, similarly like some patients and some doctors. And in that case, when you think that the number behind is not supporting this 27%, then you say that, well, it will not fly. That's why to say very clearly that the effect size of 50 or 30 or 20% of TKV growth should be qualified. And that's why I mentioned already once or several times during this workshop that what is the best way forward in this type of questions is to have the package like it was done in the GFR meeting and KFFDA EMA, and to qualify them to make it broader available for the broader application. And this is the only way to make whatever percent, 50% or 40% for the certain GFR working. Because what was important at that workshop, at least to me and my colleagues, I know similarly to the Elisa most probably, and their colleagues, that this was not a GFR which is now at the stake to say this is okay, this is not okay, but the surrogacy of GFR, which is now still based on case by case, not in general terms. That's why the only thing I could say from our perspective, because for the accelerated approval we don't have such a luxury position that if you have reasonable likely surrogate, you can go for accelerated approval. In Europe, we have a CMA, conditional marketing authorization. We have the clear benefit risk things, and that's why it would be. It can be based only on case by case, but not in general terms for every mode of action, for every, let's say, drug which is used for one or another condition in PKD. For acute effects, I think that we will have another question. Most probably, I will stop here.
Thank you, Romualdus. And you remind us of the importance of data sharing in some of what you're saying, because in order to get to that standard or to be able to get to that, we will need a lot more data, I think, in terms of other drugs' effects and on both GFR and TKV, and the importance of collecting both endpoints. Let's. I'm going to leave Elisa for the end this time, so why don't we go back straight to Ken next?
Thanks. So this is an interesting, perhaps slightly provocative question for regulators, but that's fine. What are the main considerators for a relevant effect size? I'll say I'm not an expert. I'm not a kidney expert. So what I would do is if a sponsor came to me with a proposal to develop a new drug and have a pivotal registration trial using TKV, the issue would be, okay, what does that mean, TKV? Are we talking height adjusted? Are we talking imaging, enhanced imaging? What imaging is being used? And then depending on all those details, one would look at the case they're making for a relevant effect size, and then one would review that to determine if that's reasonable to that or not. So again, the details matter quite a bit. The acute effects, of course, that's always a potential issue and often a real issue in terms of evaluating this. So obviously that has to be dealt with. And, you know, obviously we've talked about this in detail in the past on tolvaptan. So there are ways of handling acute effects. I don't think I need to go into that now, but obviously all of that has to be taken into account. I guess I'm saying it depends. But the last thing in relation to this issue I'd just like to emphasize is that over the last few years, when I received an update within PKDOC on the advanced imaging techniques, it did look promising to me. Obviously, I'm not saying I'm ready to embrace it as a surrogate, but it did seem there were promising advances. And so I definitely would encourage people to be thinking of biomarkers, and in this case, an imaging biomarker related to TKV, developing that and working toward its potential to be a surrogate. Obviously, a lot of validation work is required. And in that context, I just wonder if quantifying the effects of a snap image of advanced imaging TKV by developing, let's say, an index from 0 to 100 of the TKD progression index based on an algorithm where you take all the relevant variables from from a picture, from imaging of TKV, and determine how ADPKD is progressing in the individual patient. If people are working on that, and how promising that may be. I mean, that's just an idea that people may work on, but that's just the thought that I wanted to share.
Yeah, thank you, Ken. And that people are working on that. You know, in addition to the Mayo classification scheme, I think the work being done, including Sanofi in this case, I think, towards developing models will be very helpful in the future. Eliza, last but certainly not least.
Well, thank you. So I think, you know, maybe just to take a step back, because it sounds like we have very much an international audience. You know, in the US, we have two pathways to approval. One's traditional approval, which is essentially based on a validated surrogate endpoint, or what we refer to as a clinically meaningful endpoint, you know, a measure of how a patient feels, functions, or survives. And then we also have something called accelerated approval. And the idea of accelerated approval is the therapies being developed to treat a serious or life-threatening disease. And the endpoint that you're using is what we call a reasonably likely surrogate. So we think it's reasonably likely to predict the benefit on the outcome we're trying to prevent, but there's not, you know, as much certainty. And so in that setting, we require sponsors to do studies after the drug is approved and on the market to actually verify the benefit. And that's really the only setting in the U.S. where you can resolve issues of efficacy in the post-marketing setting, right? Because traditional approval, it's expected that at the time of approval, it's fully resolved. So if you think about how, or just to orient you to how we've thought about endpoints in ADPKD, for rare, you know, slowly progressive diseases like autosomal dominant polycystic kidney disease, we approve a therapy, you know, full approval based on effects on the rate of loss of renal function. And at least in the U.S., that was the primary basis that is served as the evidence base that supported our determination that tolvaptan was effective in slowing the rate of loss of renal function. And so, you know, that's the traditional approval. So yes, we accept a slope-based, you know, or a rate of loss of renal function type of endpoint. We just need to conclude that, you know, essentially that the magnitude of the treatment effect is going to translate into a meaningful effect on the outcome. that we're trying to prevent. And how we do that, I don't know if we have time to get into, but maybe just in the interest of time, I won't talk about how we get that. But do want to talk about TKV. So how we've used TKV, and just to take again a step back, look, if someone had a therapy that totally stopped the growth of the kidney, I could certainly imagine giving, full approval for that, right? But I think the issue is that most therapies have incremental effects, right, in terms of slowing the growth in the kidney. And so then the question comes up, so how big of an effect is a big enough effect, right? And so I think, how we view TKV is as a reasonably likely surrogate. And so then it becomes this just operational sort of practical question, well, how do I figure out what's a big enough effect? And how we've addressed that question is to say, look, I need to think about what it's actually feasible to do in the post-marketing setting to verify the benefit in terms of a trial that I could feasibly size and complete in a rapid time frame following approval, accelerated approval. And then I need to have this understanding of the quantitative relationship between how I think changes in total kidney volume are going to relate to the treatment effect on the endpoint I'm using in my trial. So in this case, the rate of loss of kidney function. And so it's really this sort of data-driven model, which CPAth has worked on or is very interested in, and certainly our colleagues from industry are working on as well. That's really how we do this, because at this point I don't know what to tell you is a clinically relevant effect on TKV, but what I can tell you is I want to make sure my trial that verifies the benefit is interpretable, and the only way to do so is to make sure it's adequately powered.
Sure.
So rephrase, if I can, just to verify my understanding before we move on to others. You need to have a reasonably likely surrogate, and then you need to have an effect on that reasonably likely surrogate to support the possibility of confirming it with a more objective outcome in a reasonable time and in a reasonable manner. So it's all about reasonableness, and that's contextual based on what you're seeing in your study, I would assume. Fair to say? And
also not just the size of the effect that you're seeing on TKV in your study, but also your modeling, this relationship that from other data that say, if I have this size effect on TKV, I think that I'm going to have this size effect on the endpoint in my trial.
Right. Yeah, and that goes exactly to what Leslie Inker had shared earlier on for both proteinuria and GFR, with I guess what this is a bubble plot in a sense, very similar to what has been presented before. So definitely appreciate that. Let me extend this to, I'm going to skip over patients for now because I'd like to hear from the trialists, but then if there are comments from patients, because I know this is a relatively technical discussion as to how they're thinking about it, that would be fine. But let me ask the trialists about how they view using TKV in clinical trials or in clinical programs, and what's the appropriate place and what's the appropriate use for TKV. Any comments?
Frank, may I speak, although I'm not officially a panelist?
You can, Ron.
All right, thank you. So when we initially brought forward TKV, it was to be used in early disease when we couldn't detect the GFR change. If I have a drug that protects GFR, I don't really care that much about TKV, unless it's related to symptoms. So again, TKV as a surrogate in early disease, I think is very useful. I'm not sure what it means in later disease.
And I just want to add that part of what the accelerated approval does is it allows the sponsor to get a drug on the market earlier and start to make money. And so it also provides an incentive if they can get an earlier readout of efficacy based on TKV. Some of what you're raising also gets to this issue that's also sort of the larger issue of the breadth of the patients that we include in the trial, and whether it's separate trials or a single trial with that breadth. But all important points.
Hey, Frank, I'd like to comment because I think this subject, I think Tess can speak to it as well, and the other gentleman, Peter, on this call. So I think so two things that you brought up, and I want to speak to what you said and what Eliza said. So one is the importance of data sharing, right? I was at a meeting 2 years ago where the subject was brought forward. And what are we really doing to address this? Because I'm seeing that as a huge barrier to innovation. I was involved with the HHS project, and we default back to dialysis. So I will raise the flag again. What can we do to accelerate data sharing? It's impacting. The other part I would say too is I would just encourage you to take a look at the good example of data sharing through the National Kidney Foundation, EMA, and I'm trying to think, and there was a lot of industry support of that. And out of that came, the surrogate endpoints. And I would encourage you to take a look at the commentary that was done from that meeting, myself and others, where we were very supportive of surrogate endpoints. So I think from my point of view, professionally speaking, is that I think many times the patients are willing to accept that risk. I've seen sometimes, and Lyra may see it differently, but I've seen some doctors not fully understanding surrogate endpoints. So that's just my 2 cents on the subject. Anyway, Tess and Peter maybe a comment as well.
Tess or Peter, feel free.
Yeah, I'm happy to comment. Just, I think this highlights something you also mentioned earlier, Frank, which is about, MRI should really be part of routine care, and the importance, therefore, of real-world evidence gathered from, years of collecting data from patients like us. I mean, so far we've had to rely on the CRISP database, but there are data sets all around the world, and if we look at the European reference networks, they're collecting a quite wide range of clinical measures. They're collecting how many numbers who've had genetic testing, then we've had MRI. And we should be looking to mine that data and bring it all together. And I know that you're looking at this within the RDCA CAP. I probably misspelled all those letters. But I think it also refers back to Kevin's comment about the data sharing. There's lots more data out there than just the great CRISP cohort.
No, appreciate that, Tess. One other aspect of this I think is important, and we really didn't touch on, is the issue of knowing enough about how a drug is affecting the endpoint to be able to appropriately interpret it. So acute effects is another part of this topic. That is very well understood, and I think progressing nicely in terms of GFR, less so perhaps for TKV. But this does mean, I think, in order to understand the effects on TKV, you need to know what the acute effects might be, which means you have to do an MRI at a baseline. And you probably have to do an MRI a number of weeks, or at most, a month or so post starting the intervention, just to make sure that you're not doing something, quick, acutely, and then measure it yearly thereafter, which was done in tempo. That creates a burden both for the sponsor that's paying for all those MRIs, but also potentially for patients that have to undergo a number of MRIs. So I'd like to hear from the patients and also from the trialists in terms of finding the right balance. Is that something you do in phase 2 early on in a small number of patients that are intensively studied? Once you clarify that there is no acute effect, you move on and do it more cost effectively later on, over a number of years potentially. Any thoughts, anyone?
Frank, thank you for that question. I'll say from the patient's perspective, first, and again, I'll only speak for myself. I would agree with the general sentiment that Dr. Perron shared in terms of if the therapy is targeting GFR and is stabilizing GFR, probably a little bit less of a concern for me about TKV, because the former is a primary concern. Regarding your question about imaging or intensive tests that may be more required if we're performing a particular intervention, I think you're right. It seems like a lot of the answers today, rightfully so, or it depends. And I think it does depend on either, the patient or the sponsoring organization. But I think this gets back to this notion of kind of over communication between sponsors and patients, which I think should be another standard of care if we're going to do that, right? Over communicate. If you're going to ask a patient to be a part of a study, which more than likely they're going to say yes to, again, just generalizations here. Perhaps over communicating and saying, look, there are the following kind of treatment paths. If you're willing to be imaged, or if you're willing to be kind of studied at a more aggressive rate, maybe then there's a subset of patients that can be targeted for such interventions. And then that allows for, as you mentioned, maybe a tighter group for the sponsoring, so that they're not paying for everybody in the study to be MRI'd twice a month, or two times every 3 months, or whatever it is. But I think in general, all of this gets back to this notion of not only data sharing for the patient education perspective, but just sharing of data for a general education perspective, right? Even simple aggregate measures of, on average, this is how long it takes for a decline, or we talk about these talking points that are often put in policy papers where you just have one or two things that on the surface seems kind of simplistic, like patients experience an average time to decline of this, or PKD patients have a water intake increase of this much, whatever the case may be. Those small little talking points for I would say on average more educated patients, like perhaps Tess and I, or Carrie, or advocates who have additional information, we probably may already know that. But in general, if you're looking to get individuals who have the disease interested in a study, sometimes those aggregate measures are very helpful, not only for people to kind of disseminate and understand very quickly, but it might be a way to get around some of the kind of privacy concerns that you have to deal with for dealing with individual level data. So getting away from some of the granularity and aggregating up might help on all accounts.
I only had one other point to add, which is look at the cost of dialysis and transplant in this patient group. We come to end stage quite young, and it's a huge cost burden, and we should be part of a big initiative. And the cost of doing an MRI every 5 years or something is minuscule compared to the cost of keeping me and Kevin alive, and others, long-term dialysis patients, long-term transplant patients.
But Tess, I just think this data sharing issue, though, is such a critical issue, because I don't think the patient community understands, one, that that's their data, right? So they have rights, and two, the urgency behind this and the benefit to patients. I'll just say that again. That is a critical issue that's limiting innovation in kidney disease.
Frank, I wanted to make a comment on your comment about maybe get an MRI at 1 month to determine an acute effect. I think those sort of determinations are best met during phase 2, because that will enable you to power and devise a study design. So, for example, both the Reprise study and the Lixivaptan trials were designed as randomized withdrawal studies based on a lot of information that was derived from Tempo and other short-term studies of tolvaptan. And, I'd sure hate to do another 3-year trial and find out there was an acute effect that we missed. So many nephrology trials are impacted by acute effects, and Leslie and Tom Green and others have figured out ways to analyze these, but it would be, I think, better to have this in hand when you're designing the pivotal trial. That's just an opinion.
And I think that I think from my perspective as well, in terms of the guidance we give sponsors, I think, Ron, you're absolutely right. You need to know this before you're in your phase 3. It's because it really very much affects how you're going to design the phase 3 trial, the endpoints, whether you need to do a comparison, which, with all patients off the therapy for some period at the end, et cetera. So it's so fundamental to the design and the endpoint and how you define it that it's good to know this in advance.
No, absolutely. So all those points are well taken. The figure that's in the slide on the left-hand side is actually a combination of data from a phase 2 trial that first discovered that there was an acute effect and then trying to apply it retrospect to tempo. We did not measure in tempo at 1 month or 2 months, but did measure in another trial. So we could sort of estimate that acute effect and account for it. And we did that in other trials when looking for trying to optimize dose or optimize formulation. Looking for acute effects might be helpful in some regards to see what a drug is doing. There's a question from the audience really directed towards the health authorities with regards to pediatric trials, because I think there's a sense that we don't have an awful lot to study children or young, very young adults with PKD in terms of finding out whether or not a drug might be effective for them or not, because GFR just won't budge at that stage because of the renal reserve. Is there any guidance in terms of how to use TKV in pediatric trials or use other biomarkers in pediatric trials to sort of supplement the data we might get from patients at later stage disease?
So I'm happy to provide comments. Maybe I'll go first and then others will talk. So, I think that this is a critical issue, and I don't know if the person who asked the comment was able to attend the discussion yesterday, but Dr. Mistry, who's a pediatric nephrologist that works on the kidney team at FDA, sort of outlined how we're currently thinking about this. So I think, essentially anytime you talk about a pediatric kidney disease, you're going to be talking incredibly rare, right? I know, we think of ADPKD as being uncommon, but I understand that, everyone has it at birth, but in terms of, being able to talk about a population, which you could already say up front is, someone who has the disease and is at high risk of progressing or at sufficient risk of progressing over their lifetime, I think you're already talking about perhaps the smaller population, right? And so I think the first question is, can you even identify who this population is? And it sounds like at least among the adolescents, we know how to do this. And so the next question is, what can I do feasibly in a clinical trial, right? Because it's such a small population. And I think the approach we've adopted, not just in ADPKD, but if you talk about any sort of kidney space, I can't think any kidney, but even if you think about if someone were developing a therapy for chronic kidney disease or something like that, essentially we're focusing on this concept of bridging biomarker. So the idea is that the biomarker, the thing that you're measuring in the blood or the urine, it's not an established surrogate, right? It doesn't have the database maybe that EGFR does. It doesn't have the evidence base that TKV does. But you at least know that in that development program in adults, that biomarker explains a significant proportion of the treatment effects seen in adults. So at least tracks for that drug, right, in the disease in adults. And so it's still a leap of faith, but it's not, perhaps an unreasonable leap of faith to say, okay, I'm going to bring that biomarker down, which is something I can measure in these kids and detect an effect on in kids. I'm going to bring that down, and that's how extrapolate. That's how I'm going to infer efficacy in children. It's a weaker database, but I think the reality is, just given the size of the population, we're often forced to accept some measure of uncertainty. So the question is always, is it at least enough evidence that I feel reasonably reassured that it will be effective? I think the other thing we usually need to know about these biomarkers when we use a bridging biomarker is that there already is some data or general data in kids to say that it probably does sort of associate in some way with outcomes, et cetera. Often we look to that as well. But yeah, that's sort of the general approach we're taking because, frankly, I don't know if you're going to be able to do studies given the number of kids out there looking at TKV as an endpoint. Maybe you can, but it just isn't clear at this point, to us at least.
Thank you, Eliza. It's good to know that at least if you can make the argument and you have some data to support that the benefit is likely being transmitted through that biomarker, even if it isn't validated, that is something that the agency would consider. I'm hoping that that's the case for Europe and Canada. If it significantly differs, please let us know. Otherwise, we'll perhaps move on to.
Could I just make a quick point? I don't have any objection at all to what Eliza said. I just want to make a quick point. Yesterday, it was mentioned onset. Well, these are pediatric patients who are rapid progressors. Time to onset of hypertensive status. I think that I've felt for a long time that could be a useful clinical outcome in as one element of measuring progression of ADPKD, and maybe especially so in the pediatric population. And it would be of interest if in the pediatric population of ADPKD who are identified as rapid progressors, if you could in a clinical trial show us a significant, apparently clinically meaningful delay in the onset of the becoming hypertensive, if that has been modeled to commensurate benefit downstream eventually on EGFR, et cetera. I think there could be some potential, at least for people to explore that. And wouldn't it be a radical idea if the modeling was strong to consider that as a surrogate for approval in that subset of patients? I'm obviously open to other biomarkers as well, and sometimes coupling that could add more power, but of course that would have to be done with care.
Thank you, Ken. And actually, that highlights that there are a large number of less clear or less dramatic changes that are observed in kids. Concentrating defects, those early changes in blood pressure, maybe in nocturnal dipping of blood pressure, et cetera. And out of Denver, a study on left ventricular hypertrophy, all of which might be indicative of early changes in this disease, cardiorenal, that might be useful. And so if regulators would consider those in kids, dependent on the effects of the drug themselves. Obviously, if you have an antihypertensive drug, it's hard to test for delay of onset of hypertension, but taking that into account, but also taking into account what Eliza said, that it makes reasonable sense that that the mechanism is related and that mechanism might translate to later outcomes would be very beneficial.
Yeah, and yesterday, just to add, albuminuria, it was mentioned that could be an important marker potentially of disease progression in the pediatric ADPKD. I've been pretty neutral about using albuminuria for an efficacy parameter in the adult population, but based on what I heard, it could be that could be another marker that could be of interest to explore.
I think there's additional gathering data on proteinuria, albuminuria, and PKD, but way more needs to be done. I'm sure Eliza would agree with that for this disease, because it's very different from the nephrotic, glomerulopathies.
Yeah. So I have a question for Ken. So a lot of blood pressure measurements in kids is based on ambulatory blood pressure measurements. That is, I don't think there's any pediatric nephrologists in the audience. If you are, you should speak up, but I believe that's the standard of care for assessing blood pressure in children. And then it's also based on the percentile range as a percentile compared to normal. So it's, I think, less straightforward, but I think there's a lot of data suggesting that blood pressure regulation in children with PKD is abnormal. So I think it would be a good thing to look at. So I hear that Jalila Macaulay is in the audience, so maybe you could unmute her and she could comment on that.
And Max as well, Max Lebo.
Well, I don't know if they're unmuted or not, but we could certainly address that in the chat.
Well, Max says ABPM is the standard of care, at least in Europe, in his written comments.
Good.
Well, I would just say I don't have any problem looking at ABPM data. I haven't to date, so I don't have any opinion. But obviously, if people are using that, that's the standard of care. And as you know, with ABPM, there's different analyses that you can or should do, probably more than one. So obviously, all that has to be worked out as what's most relevant in this population. But assuming that one can justify and validate its use, I'm certainly open to looking at that. I think it's an intriguing possibility.
Great. So one of our later slides talks about pediatric issues. If we're having difficulty getting people unmuted, maybe by then we would be able to do that. We're getting a lot of written comments from a whole cohort of pediatric nephrologists that are something on sort of this and other endpoints. So let's reassure them that we'll be able to get there and then have them raise their comments when we talk a little bit more about pediatric endpoints.
Thank you all.
On. Yeah, thank you. Let's move on to the next question, though, having to do with the measurement of the renal outcome endpoint. And I just remind everyone, and Lisa, I think, has already alluded to this, tolvaptan was approved on the basis of delta in GFR, not in the standard composite endpoint that's listed here. And I don't know if that is set a standard for everyone else, or if there's still value. And this is not just for regulators, but for the academicians, the clinicians, and the patients. If there's value in being able to quantify or understand what degree of delay might be achieved in putting off end-stage kidney disease. Any comments from anyone on this question? Because I don't believe, based on tolvaptan's approval, that it's absolutely necessary to have this endpoint, but it might still be advisable. And I don't know, do any of the regulators want to address that initially?
I'll just throw out there that you know, this was the endpoint that was historically used, I think, more in the diabetic kidney disease space. This is a rare disease, and I think, fortunately, work done by others would indicate, you know, Leslie, et cetera, you know, I think gives us confidence in the eGFR slope-based endpoint. I think, you know, also just want to highlight that it's not. I think the idea, too, is that we need to believe the effect size seen on GFR in the study is clinically meaningful. And how we do that is by getting data from the trial or development program that shows that the effect continues to accrue over time across the various stages of disease, which is why, at least in the U.S., with tolvaptan, one of the reasons why we wanted the second trial in patients with GFRs less than 60. I think when tolvaptan was initially studied, there was this hypothesis that it would only work in patients who had preserved GFR, but that left a lot of outstanding questions as well about, okay, what happens when you progress, and you know, how do I actually understand the nature of the benefit in terms of progression to kidney failure if all I have is this early snapshot of its effect at an early stage of disease. So we generally wanted to see data across the stages of disease to understand.
And Elisa, I think that snapshot approach, I think that's the term you've used in the past of understanding early and late disease, may be another argument for not being overly restrictive in terms of the population studied, and being careful to try to measure GFR even in those patients whose GFR is not changing because they're very early in disease, but maybe over a longer period of time or with more careful measurements show that there are benefits early as well as late, and that gives you that continuum of potentially cumulative benefit, right?
I could say, from my point of view, having this as a composite endpoint in every single ADPK trial, I don't think is necessary or appropriate. If you're, but I'm talking about the composite endpoint as listed. If you have very early stage disease, you're not likely to get many events. However, what I think is very important from my point of view is each of these components be captured in a rigorous way. They don't have to be adjudicated, but they have to be captured. So even at an early stage trial, we want to know if people are going into renal failure. Obviously, that's going to be probably a very rare event, but we need to capture that with rigor, renal death. And then the other point to emphasize is EGFR has, in my mind, it's very important that it be monitored over time for any chronic trial in ADPKD as an endpoint. Now, you know, if it's early stage, it's not going to be probably a surrogate or involved. It probably doesn't make any sense, but I think it needs to be a secondary endpoint. And basically what I need to see is a time course of EGFR and maybe subgroup analysis as appropriate. So any trial, I think, needs to be looking serially at EGFR in ADPKD.
Thank you. Any comments from the trialists or from patients with regard to, should we be studying this just from the perspective of, you know, comparing it to other treatments in diabetic kidney disease or other forms of CKD?
You know, Frank, the only thought I was just thinking about is like secondary endpoints, like patient-reported outcome measures. I don't know if that fits in this discussion here, but I guess where I'm coming from is that there's some, you know, thinking on the other part when you do bring a treatment to the marketplace, there's some school of thought of saying, well, what's the value of having 3 years of that native kidney? And some people are under the false impression that, you know, you go to transplant, all your problems are taken care of, which is, you know, not the case. So I'm just wondering, that's the part I look at, like cognition, for example. You know, if there's ways to provide additional, proving the benefit of slowing this down, that's something I guess I would be interested to explore.
Actually, that's a good point, Kevin. And I don't know if we have Dorothy here, but my recollection of the data on the PROs that were collected in the observational program that actually did go all the way to those patients who were on or approaching dialysis or had already been transplanted. My recollection is that instrument was very sensitive in terms of determining that you actually did have increases in physical and emotional stresses as you approach that time frame. I'm not sure exactly what the inflection point is, but there were also increases in healthcare costs as you got to that point. So knowing that a drug not only delays your getting there, but maybe it could even help patients that are already there for the few years they might be there might be useful. And Dorothy, if you did want to comment, I'm told you are now unmuted.
I'm still here. We did indeed look at that in the Overture study as well, since we didn't collect the information in our pivotal trials, but we do have some of that data from Overture. And as you said, Frank, it's really across the disease stages. So it's from CKD1 all the way through CKD5. And I think we have up to, I think in some patients, up to 3 years worth of follow-up data there. It's not something that we've explored in detail, I think, but there is some indication that there are some, you know, as you progress through the disease stages, some of that does change.
Yeah,
where, you know, you have some indication even so that there's a lot more anxiety, for example, in the earlier disease stages where patients aren't quite as sure on their trajectory of progression. And as you progress through disease stages, I think that goes down because you have more certainty about where you're going to end up.
That's exactly it. And I think that there is a lot of value, this is just my opinion, but exploring that, because I still don't, I'll say this again, I don't think people really understand the full burden of kidney disease. So
thank you, Kevin. Yeah, and I think this question may be, you know, not only on how to design and make sure that you capture all relevant endpoints, but also all relevant stages of disease. Are there any other comments or questions on this? I know now I think I am running up against a time constraint, so I want to make sure we address some of the last questions, including the ones for the pediatric population. Any other questions or comments?
Yes, if I may, just to say that in Europe we have our guideline, which I was rapporteur for that. That's why maybe I'm biased, but we always highlight that these all-cause renal failure things should be always reported. And of course, in real life, and in Tolvaptan case, it was not informative reporting, but I fully agree with everyone that it depends on the early, late, advanced disease stage and when you can expect. But at least we should always put this endpoint into the set of the secondary endpoints.
Thank you, Romualdus. And I recall that there were discussions, for example, about following, even if your treatment study is for only 3 or so years, and then everyone goes on to active therapy and an extension trial or what have you, that maybe if you actually did follow those patients all the way to end-stage kidney disease, you might see some difference between the early and late treatment. I think it may be useful, particularly if you're already studying patients that are late, to capture that data. Let me move on to the next question, which speaks a little bit about the acute and the potential of acute effects on either GFR or TKV. And as I said earlier, I think we know much more about this for GFR and how to account for it. I'm presuming that the principles are similar, if not the same, but really want to understand what the perspective is from regulators as to what kind of data, what kind of information, what kind of analyses might be most useful for studying acute effects, whether it's the chronic slope approach, looking pre-treatment to post-treatment, after a treatment withdrawal, for example, and of course, clarifying what's the underlying cause of those acute effects early on, I think, as we've already discussed in phase 2. Are there any comments from anyone on that? And I don't know if Leslie is also available, but there may be an opinion from CKD-EPI as to the best method.
And maybe I'll just start by just saying that, I think one issue here is sort of when in development do you want to understand this issue? And I think you want to understand whether your therapy has an acute effect before your phase 3 registration trial. But I think the other question you're raising is when you actually know that you have an acute effect, how do you analyze the data, right? And I think, I assume chronic slope, you're probably referring to the eGFR chronic slope. Maybe you're also referring to it for TKV. I think, we've been wary of chronic slopes because it requires actually a whole lot of assumptions, which can't be validated, and there's concern for bias. So typically when someone has an acute effect on eGFR, and they're trying to deal with it in their study, we recommend the pre-treatment to post-treatment difference. That's sort of our our first recommendation. And Frank, I think you're smiling because I think we've engaged in these discussions before.
Mm-hmm. Absolutely, but it's good to get this out in the open, isn't it? Romualdus, do you have any thoughts from a European perspective?
As we discussed so many times, whether chronic slope is acting, can be used for whatever reason. And I don't recall any case in advisors in a discussion that we agreed that the chronic slope is sufficient enough. It is promising. It is maybe very good for having a sense of what's going on in the long term, but not for the primary analysis. So we always for the total slopes, and of course on top of that, of course the main question is the nature, magnitude, duration, reversibility of the acute effect. This is what matters, and how to resolve that is, of course, phase 2 study is the best place where to analyze and to understand whether what happened after withdrawal of the product and the magnitude of the acute effect, how it will impact on the therapeutic thinking, should we do? Should we stop the stop the drug, or continue, because we at the end of the day will be have a beneficial total final outcome? And I think the patients, they are also not the last who will say that this is important or not important. So coming for the GFR, these small changes in few mLs, if we speak about the GFR, 5 mLs, 10 mLs for a short period of time, of course, doesn't matter. So, but it might be different if the acute effect is much stronger. So we have a variety of the magnitudes, durations, and ignore them, and to say that chronic slope is very convenient for the sample size estimation, whatever, it is very difficult to accept. So at least in Europe, we never accept it.
Thank you, Romualdus. Let me just add one comment from a practical perspective, and I want to get the patient feedback as well. When Reprise was being designed as a study, in this case, in close collaboration with Elisa and the group from FDA, we knew we wanted to collect pre and post-treatment measurements of GFR or blood samples relatively regularly, but spaced appropriately apart, and then quickly, during a short period of time after treatment had been discontinued, to verify where patients ended up after treatment was removed. And with regard to decreasing variability, there was a lot of conversation about, are 3 samples enough? Shouldn't we get 5 samples of blood within a 2-week period or something like that? And our steering committee actually said, whoa, if I were a patient, I'm not sure I'd want to go into this study. So I'd like to hear the patient perspective. If you know that this is a real important issue for scientists and regulators to understand whether the drug is really having a good and persistent beneficial effect, are 5 blood draws within a 2-week period too much? Anyone?
I agree with you, and this is difficult issue if you have if you base your conclusions based on this reversibility things. I think that when you have a longer duration of the study, you will be clear that whatever rebound will be happening or not at all, you are still above the control arm, so you don't need that. I think that the issue is when you have a very short period of the study, then you might enter into these discussions and then all these challenges.
Granted, Romualdus, absolutely. Reprise was 1 year, and so you had to measure the delta over 1 year of treatment as opposed to Tempo, which was 3. So yes, your alternative is to stick a patient 5 times or 3 times, what have you, in a short period of time, or make them tolerate the study for 3 years or so. It's sort of one or the other. But from a patient perspective, we sometimes seem to ask an awful lot, whether it's blood collection, urine collection, repeated MRIs, asking every week on a PRO. I really like to understand from your perspective, if we communicate, and this is to Peter's point, if we communicate how important it is it a problem for you?
It's Tess. I think I'm
Go ahead,
with
Tess.
Kevin. Well, I guess it depends what you use by sticking. I mean, if it's traditional bloodletting, then it's too much from a personal perspective. But there are these new point of care creatinine testing devices that are being approved, certainly approved in the UK now, which are being used for. In fact, it has been suggested that they could be used if they can be tested for liver enzymes for post. Checking liver enzymes at home with tolvaptan, but certainly used in pregnant ladies and increasingly in post-transplant patients for a quick creatinine test. I don't think that would be an issue, but going in for some bloodletting at that frequency would be a lot. On the other hand, people are concerned by that acute drop. They always mention it in the groups. It comes up all the time, and they usually get reassured by the other patients that it's short-term reversible, blah, blah, blah.
Yeah, thank you, Ted.
See, I guess here's the
to use the word jab, which I think is more appropriate for the UK, getting a jab as opposed to a stick, right?
Well, yeah, it's equally horrible.
Yeah, Frank, here's what I look at it as, is that I guess the question I see is that when clinical trials really are designed for everyone but the patients who are in the trials. I mean, if we're going to be honest here. And so I look at the example like with telehealth, right? So why didn't that come out earlier? Why did COVID force that, right? So I guess my sense is that if things are being done to minimize the burden upon the person, like if you're going to have labs come into their home, then it's less of a burden. But if you're putting more burdens upon the person, I think that's where I would just say my overall comment is I don't think clinical trials are designed with the patient in mind, logistically.
No, thank you, Kevin. I think
I say it's, yeah, I just say we're ripe for disruption. I'll just say that.
Yeah, no, I think we're starting to listen, and I think COVID is probably helping in terms of doing things a little bit easier from the patient's perspective, and probably will be easier from our perspective as well as trialists. Any other comments? I wanted to move on because we are definitely into John Michael's time, even though he was gracious enough to allow us to do this.
We still aren't able to get MRIs remotely.
Granted.
There's an app for that. There's an app for that now.
Well, there's an app for just about anything. I have a strong bias that I think PROs should be collected in real time on a smart device, not based on some arbitrary recall period. I mean, I can't remember what I had for breakfast yesterday. How am I going to remember how much pain I had 4 weeks ago? So, you know,
there's a wearable for that.
What's that?
There's a wearable for that.
Yes.
Talking about biases and PROs, I just would like to table my interest in PROs and also composites. We don't seem to have time to go into the case for using composites of clinical outcomes, but just food for thought. I think it would advance the field if regulators would have an expectation that for any registration trial in ADPKD, that at least some credible composite of clinical outcomes be included as a secondary endpoint. So I'm not suggesting it would be a surrogate for approval unless it was fully validated, of course, but also a pro, right? Why aren't we doing this? We should be including the best pro on a per study basis in every registration trial as a secondary endpoint. Because when you do this, then you learn from that. You get data, but you also learn how to improve them. So that's just something I wanted to table for the attention of the regulators.
So listen, someone is giving me a very subtle hint by moving me to the wrap-up slide that we need to wrap up. So before we do that, let me just skip through the last questions that we didn't get to and promise that somehow or other we will continue to solicit advice on that. So we ended up here. We were hoping to talk about demands and protecting against missing data. We weren't able to do that necessarily, but there was a conversation, and we would look to further advice with regard to that. We talked very briefly early on about using a broader population to try to understand safety and efficacy better for all patients, not just those that are likely to be enriched for a particular endpoint. And then we were hoping to speak more to pediatric considerations and whether or not you needed to show benefits before moving to pediatric patients or whether you could study them concurrently. I think the standard is that we would want to see some likelihood of a benefit before including patients in a pediatric age group. But we clearly want to include them, and we want to treat them appropriately because we have to understand that school-age children, children in general, may have difficulty maintaining compliance and really understanding fully the importance of their involvement in the trials. So we've answered a lot of questions already from the audience, certainly not all that we could. I apologize for that for anyone who has posted questions or wanted to raise their hand. And in wrap up for this session, I'd just like to thank all of those who participated. The PKD Outcomes Consortium, I think, is taking all of this very seriously. We'd like to understand whether there are next steps, whether that is creating a publication or a consensus paper of some sort, or actually working with various agencies or various companies toward developing these standards, whether it's adopting CDISC to CRFs or actually creating template protocols that might be appropriate for phase 2 or phase 3. We're very serious about finding the right tools to accelerate research in PKD and accelerate the development of more therapeutically useful drugs. So with that, I'd like to, again, thank everyone for their active participation and ask John Michael if he would like to move on to the final stages of our 2-day meeting.
Great, thanks a lot, Frank. Wendy, if you could go to the next slide. I'd just like to echo Frank's statements about thank you everyone for your participation and listening to the sessions. I know it got very technical at some points, but that's what we needed as a community. This last section, super technical, super clinical, but the things that were said were really important between regulators and academic scientists and industry scientists. So I think we did what we needed to do, right? And I really appreciate everyone's candidness. I think we could have just had a panel session for the next 8 hours, and I still don't think we would have gone through all these topics. So we still have a lot of work to do, as we've talked about. We're going to be looking for a publication, looking forward to a series of publications around this. So I'd like to thank everybody. Next slide, please. Just a quick plug here for folks that are interested in more about the kidney. We're going to be talking about drug-induced kidney injury at a biomarkers next Wednesday. If folks are interested, it's free to tune into. Please tune in. These may be biomarkers that we can actually take advantage of within the polycystic kidney disease space. So that's something to think about. But with that, I'd like to thank you all. Have a great day, and I appreciate the great participation. Bye-bye, everybody.